I'm just waiting when a number of participants will go onto the platform.
Good morning to our Russian colleagues. Neelam here from Kuala Lumpur, Malaysia.
Hello, Neelam. Good to hear you.
Yeah. Thank you very much, Professor Zaitun and Dr. Firdaus for participating. I think we should be starting soon, Professor Lisitsa.
Yes, we do. We are just waiting when the number of participants will go onto the plateau, because people start to their connections at 9 o'clock. Some people start, and I just looking at every 20 seconds a new participant join us, and I mean in 1 minute we will start.
I think for Malaysians we don't have an issue because it's already 2 o'clock in the afternoon here.
Okay, so today we start our one-day online roundtable on bioinformatics. And first of all, I would like to thank Neelam and Maya for their efforts in organizing this event. And then I would like to ask colleagues to take their coffees, teas, and snacks and whatever. We will spend 4 hours without break, but our main purpose is to acquaint each other which are the directions developed in the universities of Malaysia and in the research institutions of Russia, with a special focus on the bioinformatics. And I thank you, colleagues from Malaysia and from Russia, for the time and contribution. And I think that it is a good start for the joint virtual and later face-to-face activities. So we well know Zoom scenario, then we switch off the mics if someone is talking, not to interfere, and we sometimes it makes sense to switch camera if we could see our faces, but during the translation of the presentation I recommend to switch camera off, not to be heavy on the channel. I kindly asked to co-chair the event Professor Maud Firdaus Reich. Maybe you know him quite well, but anyway, I feel my responsibility to introduce him. As a professor. I've forgotten to switch on video. Sorry. Professor of Bioinformatics and Computational Structural Biology, working in UTM Malaysia, that is Kuala Lumpur. And I appreciate an opportunity to round table with Professor Reich, and we suggest that I will announce Russian speakers, and Professor Reich will announce Malaysian speakers, not to be correct in pronouncing the names. That's how we will work. And I give some introductory word to Professor Reich. How are you?
Fine. Good morning. Good morning, everyone, and good afternoon, everyone in Malaysia.
Good. So we can start our agenda, and I introduce Professor Reich as the first speaker on the structural biology. Please, Professor, stage is yours.
Right. Good morning again to everyone in Moscow and Russia, and good afternoon to everyone here in Malaysia. Thank you to the organizers for giving me a bit of an opportunity to talk and discuss some of the work that I've been doing in my lab for slightly over a decade. And I think the idea or the discussion for having this online roundtable started when Andre and his colleagues came over to Malaysia and visited me on the 17th February last 2 months ago. And I think you also went to a few other universities, including UTM, of which there are representatives here. Okay, so I want to begin my talk with this very general statement that we all more or less are aware of. The functions of biological macromolecules are determined by the three-dimensional structures that they take up or fold into. And this is a sampling of the diversity of structures that proteins and RNA can take up in order to carry out their functions. The overall structure of proteins and nucleic acids we know are important for their functions, but it is only a subset of residues that are directly involved in a particular function mechanism. And these functional residues are usually conserved as 3D substructures, while the rest of the fold can be different. So if we find 3D substructures with known mechanisms in structures with unknown functions, therefore the mechanisms that these substructures have may be involved in could be similar. I want to take you through this example, the example of this concept here and demonstrate it by using these two enzymes, chymotrypsin and certylysin. As you can see on the left and on the right, the arrangement of these three amino acid residues, aspartate, histidine, and serine, are very similar, despite chymotrypsin and certylysin being enzymes of different ancestry. That means they are not related to each other. They are not homologous to each other. They have no similar evolutionary ancestor. So different enzymes can carry out catalysis using the same reaction mechanism, even though they are not related. And in this work, I think I've mentioned the word or the term substructures, 3D substructures. And what I'm referring to is basically just constellations of residues that are components of a larger macromolecule, either a protein or an RNA structure. And although it is also sometimes referred to as 3D motifs and structural motifs, when we term it as a motif, it implies repetition, but that's not always the case, because in some cases, it's a discovery of an arrangement that is not necessarily something that we see in other examples. So until 2020, the accuracy of protein structure prediction or de novo structure prediction was rather low, and it was quite inconsistent. But recently, the methods that utilize artificial intelligence have proven to be quite successful. And two notable algorithms are Rosetta Fold and AlphaFold. And in July of 2021, AlphaFold computed about almost 400,000 structures for 21 model organisms commonly used in biology. And a year later, in 2022, the same group at the European Bioinformatics Institute computed the structures of more than 200 million records that are available in the UniProt database. Now let's compare that to what's available in the Protein Data Bank, which is the database containing records of coordinates for protein structures that are experimentally solved. Now, in the Protein Data Bank, there are about only slightly over 200,000 records, and that's after 52 years of being in operation. And many of these structures have unknown or uncharacterized function. As you can see from a rough calculation here, generally about 20% of those that are predicted by AlphaFold in the UniProt as well as in the model organism data sets are labeled as having uncharacterized function. And for the PDB, roughly about 9% have unknown function or are labeled or annotated as having unknown function. But in addition to protein structures that have unknown functions, some proteins may actually have known functions, but have an unknown mechanism. For example, we may know a protein to be a nuclease from DNA assays, but it may not be known how that nuclease is actually cleaving the DNA. So the mechanism might be unknown. So using methods like AlphaFold, we now have more new structures to analyze and understand, and less effort is actually spent on trying to predict and getting the right structures. So this allows us to spend more time on analysis and understanding the functions of these structures. So how do we go about assigning functions to this flood of structured data? Obviously, we can experiment and assay them, but where do we start? What assay do we start with? So can we take a substructure associated with a known function and find similar ones in structures with unknown functions? And our work, the work in my lab, has shown that this is actually possible. So the similar substructure in uncharacterized structures implies that it may share a similar mechanism or a similar role to that in the structure with known function. Although the overall function of the two proteins might be different. So it might have a similar binding site, but the rest of the proteins may be doing different things or contribute to different overall functions. And how we're doing that is representing the amino acid side chains as graphs, or what we call pseudoatom vector graphs. So I'm using this schematic here to demonstrate that these points in yellow and green are basically what we term as pseudoatoms, and they can form the nodes of a graph, while the distances between these pseudoatoms are the edges of a graph. And we can then translate that into distant matrices, and we can then use subgraph isomorphism algorithms to actually find similarities or subgraph isomorphism similarities. That seems like an obvious problem and an obvious solution, which it is. But what we are doing differently is the fact that we are actually using the side chain of the amino acid as opposed to many other programs that are using the C-alpha backbone. So there are advantages to using the side chains, as I am demonstrating here in this graphic, where you can clearly see that the C-alphas there not superimposed to each other, while the side chains are actually superposed or are actually aligned to each other. And this is advantageous if we're searching for functional residues, because it is the side chains that are the business end of these amino acids. So we've developed those algorithms and we serve them as programs, web applications that the scientific community can use. For example, this one, it takes a whole PDB structure and it searches motif databases, or it can take a motif and searches a structured database or data sets, including the AlphaFold data sets. So what else can we do with such a searching capacity? Let's say we know a known drug binding site, okay? Can we find similar binding sites in structures that are not known to bind to that particular drug compound? And our searches reveal that yes, we can actually do that. And during the SARS-CoV-2 pandemic, or the COVID-19 pandemic, we actually did this analysis where we identified SARS-CoV-2 proteins that could be targeted with existing or available compounds. And some of these have been proven in clinical trials, and some of these are actually used clinically. And in this example, we show that the SARS-CoV-2 ADP ribose phosphatase has actually multiple sites that could bind to different drugs. So if we can find that, it's possible that these compounds could also bind to human proteins as off-target sites. So that could lead to unintended side effects or toxicity. Having said that, it doesn't necessarily mean that those compounds cannot be used, because technically these are drugs that had already been approved for use. So it doesn't discount those compounds for use as therapeutics, but what it can do is it provides a clinical trials with the intelligence that if they're observing some particular side effect, and they might be able to attribute a particular mechanism to that side effect. And of course, we expect this capability to improve as more structural data becomes available. So what else can we do with such a capacity? This is one example that is actually of other people using our algorithm, and they have identified a novel motif of 4 aspartic acid clusters occurring in different enzymes. But is this useful for computed models such as those made by AlphaFold? So in this particular example here, I'm showing proteins from this family called Bacteria lethal factor 1. And on the right here, what I'm showing is the superposition of the active site for this BLF1 protein from two examples that are very remotely associated. So they're quite divergent in terms of sequence identity, where this only got 19% sequence identity, but AlphaFold has managed to model it quite accurately and it forms the active site quite well. But in this example in the middle, AlphaFold was not able to model it very well, as denoted by the confidence PLDDT scores given by AlphaFold. So AlphaFold doesn't consider this a very good model. And although these three residues also are present in this particular model, it's not able to be superposed to the known active site. So it shows that it's useful, especially when you get the right structure. Okay. What if we don't have an exact idea of what we're searching for? Can we still find something? So in this particular case, we're showing a computer program that searches for a conceptual arrangement. So you don't need to know what you're looking for. But let's say if you have an idea, oh, I want to search for a cysteine, histidine pairing, for example. And in this case, we did a conceptual search for such a pairing, and we were able to find this in two unrelated structures, as you can see here. So in gold is one structure, and in blue is one structure. And you can see that this metal binding site actually superposes quite well, although these two structures are quite different from each other. And we've been able to use this to find motifs that are present or understood only in a 3D context. And in some cases, there are single residues at the sequence level. So it's not something that will be detectable in a sequence alignment. So many of these are what we term as novel motifs that are hiding in plain sight. And can we then also use such an approach for RNA structures? And the answer to that is yes. So because we're seeing an increase in RNA structures being deposited in the PDB, we decided to see whether we could use the same methods or the same approach, whether it's applicable for finding clusters of RNA bases. And we were able to use that same approach, and we were able to find novel RNA base arrangements. And we also explored whether we could find or we could use this concept of drug repositioning for drugs that target RNA molecules. And our analysis showed that yes, it can be done, but it is not as clear-cut as it might be for protein. So I'll be happy to discuss this if anyone is curious about it. So in general, we have developed these approaches that we serve to the general scientific community as these tools, and they can do different things. They take different inputs, search different databases, both for proteins and also for RNA structures. But for many users, it's confusing as to which particular tool should be used when. So as a solution, we develop a one site that can interface all of these programs, and the user basically is guided through the process up to the point of having their particular question about their structure answered. So they don't really need to know which particular program to use. The program or the interface basically guides them through that process up to the point they would have their questions answered. So in conclusion, 3D substructure searches in biological macromolecules, both protein and nucleic acids, can be carried out by representing the amino acid side chains or bases as graphs. And such searches can allow for 3D substructures to be found in a database of structures or vice versa. And the increasing availability of protein and RNA structures, I think, will be the next frontier towards understanding the extent of atomic level mechanistic diversity and conservation. We want to be able to see how conserved some mechanisms are despite the fact that they are from different proteins. And we hope that by we will be able to assign more functions and of course discover new biology, and of course having lots more fun in that process. I think before I end, I would like to thank all the postgraduate students who have contributed to the work that I presented just now, and also to the funding I've received over the years, and my various collaborators who have contributed bits and pieces to the work that I presented just now. And I think I will end there. Thank you very much.
Thank you very much, Professor Wright, for presenting a family of very important bioinformatic programs which manage a new approach to match the motifs and to make this functional annotation and even drug repositioning. Very promising task we also do with that. Colleagues, the format of Roundtable is because we don't have time to ask questions, but please don't hesitate to put them in your records, and then through the organizing committee, contact to any of the speakers you are interested and detailize what is should be understood. That is a kind we work and we move further. And I represent Professor Olga Galzitskaya, next speaker. She is from Institute of Protein, Pushchina, that is nearby the Moscow, small city which is almost devoted to the structural biology. And Professor Galzitskaya, she is in charge of the laboratory of proteomics and bioinformatics. We continue the topic of the structural biology with this report. Olga?
Oksana.
Oh, Oksana, sorry.
Okay. Good morning, everybody, and thank you for invitation to give talk on this roundtable. And today I want to talk you about some hot topics in bioinformatics and maybe molecular biology, and to say about such a problem as my talk was titled "To be folded, to be unfolded, or to be aggregated" on the example of the ribosomal protein S1 and its fragments. If we want to start some project, and this project will connect with some protein, and we want to find some functions of our protein, or want to increase enzymatic activity, or to find new properties, we should get some dossier for our proteins. So in the last century, this scheme was very important for the process of protein folding, and especially these intermediate states was focused on many researchers, because this state is beginning for formation of such states as amyloid fibrils, and also formation of disordered aggregates. But in new century appear more complicated scheme, and in this scheme appear new states and new transitions. For example, you can look such important and now hot topics in molecular biology, liquid gel phase transition. And what we know about these transitions, that for such transition, it takes parts for formation of stress granules, ribonucleic complexes, p-bodies, and other complexes. Also, what is necessary for protein, what structure should be to form such complexes. And scientists found that this protein, for example, should have disordered regions. And one of good example is prime-like domain. This is prime-like domain usually has disordered structure, and so it's possible to do such reversible transition, liquid in gel. Also in this diagram appear many important states, such pathological aggregates in the form of amyloid fibrils. Also we know that these states, amyloid fibril, may be functional, because we know that many organisms in human body and in plants have amyloids as functional amyloids. So we in our laboratory developed several programs. Now I will describe several programs for description of protein structure. And we elaborated programs for prediction of disordered regions and also amyloidogenic regions. This is regions responsible for formation of disordered and amyloid states. Intrinsically disordered proteins have no three-dimensional structure, and it's important that this protein have many functions and many partners in interactions. It's example of prime domains. Also, we use when we begin to elaborate our programs, we use some physical ideas, and sometimes using physics, it's also necessary to elaborate new programs. And the idea is that if we have sufficient number of contexts in the protein, we will have global states. If we have no sufficient context, will be disordered states. And if such context, expected context will be large, so appear amyloid states. Later, using physical models such as Ising model, we elaborated new programs, Ising-Struct, for prediction of disordered state, and our program was included in the top of methods for prediction of disordered regions or disordered proteins. It's special now. There are some competitions, height prediction, and people estimated the possibility of different program and different methods. So in my talk, I will consider one of important protein, ribosomal S1 protein, and maybe describe some important function of this protein and how we can suggest some hypotheses and to check and to obtain some very important results. This is important structure because this is ribosomal S1 protein, but this protein present only in bacterial and archaean domain of life. And what is interesting that this domain have repetition of domains and number this is domains, maybe from 1 changes from 1 to 6. And how many domains we can conclude what phylum bacterium we have. So number of domains can send you what bacterium you have, because gram-negative bacteria have only 6 domains in this protein. Also, this protein has many disordered loops and maybe some disordered region in domains, because until now it's not possible to receive full X-ray structure for this protein. Only some domain was determined using NMR technique. So when we study some protein, also it's necessary to consider some homologs, because we want to know history of this protein, evolution, how conserve our protein, and in this case it's complicated protein, and we want to know what domains will be more conservative, and we consider this more than 1,000 of sequences, try to answer this question, what domain is more conservative. And for large domains, for example, for 6 domains, central part, it's more conserved because we obtain more high identity between different S1 ribosomal proteins. Also, we calculated, I described about intrinsically disordered regions. Also, we may say about intrinsically flexibility regions. Some domains more flexible than others. And what is important also to look on the prediction of amyloidogenic regions, and what is important that one domain is very hard protein, and later you will see why hard, because this sequence will include many amyloidogenic regions. So we obtain some bioinformatic dossier about this protein, and maybe was asked about is it possible to produce amyloid fibrils for this protein. And this is prediction. Usually I want to underline this each domain in this complicated protein has different functions, but until now people know all functions from domains and only can study a model organism, E. coli and Thermophilus. And this is example for prediction of amyloidogenic regions using different programs. Usually we know in bioinformatics that it's better to use several programs to find some consensus what you want, to find some docking region or disordered or amyloidogenic. It's better to find some consensus. This is example of prediction disordered regions for this protein. Last domain should be 6 domain, but last domain for Thermophilus with 1 protein is disordered. Well, this is prediction using our program is unstruct, and this prediction using AlphaFold. 5 domains and last is disordered. This 1 domain, you can see that this protein includes, I tell you that very hard protein because practically covered all amyloidogenic regions. And we see difference between amino acid composition of proteins from different organisms. This is from Mycoplasma mobile, S1 present as 1 domain, and Thermophilus present 5 domains, and 6 domain is disordered. So we ask, will be this protein S1 with 5 domains and protein with 1 domain form amyloid fibrils? And we not obtain this as fibrils. But next question, will be form amyloid fibrils amyloidogenic regions? But and this we can ask what we can use such properties as amyloidogenic or amyloids in the problem of molecular biology. And one of the big problem now, we know that antibiotic resistance and produce and suggestion of new antimicrobial peptides maybe will be a new solution for a pair of instead of antibiotics. And was we suggested such ideas that use amyloidogenic regions in the process of co-aggregation. If our amyloidogenic regions, these peptides, will interact with target protein, so we will have the process of co-aggregation. It means that our protein will lose protein function and we will observe some antibacterial action. So you remember that full protein not form amyloid fibrils, but if we synthesize our amyloidogenic regions predicted using consensus for programs, we synthesize this as peptides and demonstrated this is region where they correspond in structure, it's beta strands. All peptides form amyloid fibrils. Also, we demonstrated that we observe amyloid fibrils in the process of co-aggregation because protein not form amyloid fibrils, but our idea that was use peptide as agent for aggregation. So we check, this is model organism Thermophilus, antibacterial activity for these peptides and found that some peptides have minimal inhibitory concentration similar for antibiotics. But more important that we use this not only for model organism, but consider such pathogenic organism as Pseudomonas aeruginosa and Staphylococcus aureus and produce antibacterial peptides and the action of these peptides similar antibiotics. So I want to conclude in my short talk that using some bioinformatics approach, you can decide many interesting tasks, and we assume that peptides capable of co-aggregation may have intracellular targets. Interaction with each leads to cell death. The ability of artificially synthesized peptides based on the S1 ribosomal protein to manifest aggregation and antibacterial properties has been theoretically predicted and experimentally confirmed. Also important that we produce antibacterial peptides for such pathogenic organism, and also I have no time that we produce some bioinformatics servers and using such now modern deep learning approaches for production of service for prediction of amyloidogenic properties for amino acid sequences. Today, it's my finish, and I want to acknowledge people who take part in different parts of this work, because it includes theoretical, structural biology, bioinformatician, and experimental part of this work. Thank you for your attention.
Thank you very much, Oksana, and so colleagues from Professor Galzitskaya, we just have heard and information about unstructural regions in peptide structure, which always make me nervous when I do explore the results from AlphaFold, and I see a big region which is don't have any structure, and it's well to know that we do have tools that can help us to uncover the message which is behind these unstructured regions. So, Professor Reich, I give a word to you to introduce our next speaker.
All right, thank you, Andre. For the next speaker, I invite Dr. Noraza Basa from University Technology Malaysia. Dr. Noraza, you have the floor.
Okay, thank you very much to the chairperson. And thanks for the opportunity to join a one-day online roundtable on bioinformatics. So I am Noraza Basa from the Department of Chemistry, Faculty of Science, UTM. My background is actually in chemistry, but we are focusing on medicinal herbs, not only from Malaysia, but also from other countries, which include our collaborators from Nigeria, India, Bangladesh, and so on. So the topic that I will be presenting today is in the in silico evaluation of phytoconstituents from cassia species. But the in silico will be only a small part of this presentation, but throughout the process to get the compound, I will show you during this presentation. Okay, so a little brief information regarding the medicinal plant in Malaysia. There are at least about 250,000 species of flowering plants in the world. And in Southeast Asia alone, it has about 35,000. And around that, 8,000 are found in Malaysia. And until now, at least 654 species have been reported as endemic to Malaysia. And from there, about 6,000 of the floral species have been reported to possess medicinal values in the tropics. And from that total, 1,230 species have been recorded in Malaysia as medicinal plants. So there are quite a number of families, medicinal plant families in Malaysia, but my research group is focusing on some of the families, which are Culiaceae, Moringaceae, Rutaceae, Thymilaceae, Gingibraceae, and Fabaceae. So from the list of plants, it's just a brief information on some of the genera and species that we work on in our lab, which the first family is Culiaceae. It has about 49 Garcinia species in Malaysia, okay? And some of the phytochemicals reported are triterpenes, xanthone, bioflavonoids, and benzophenone. And it has variety of traditional medicine system use, such as to treat bronchitis, throat infections, and cough. Okay, so this is some of the compounds that has been isolated from our group, focusing on Garcinia parvifolia species. Okay, as from the structure, we can see there are quite numbers of xanthone isolated from the Garcinia parvifolia. Continuation with the Garcinia species, we also focus on the Garcinia hombruniana, And the different in terms of compounds isolated, as we can see here, the types of compound are more on the triterpenes compound. Move to the third species is Garcinia mangostana, which consists a lot of xanthone in a major numbers, and the compound is alpha mangosteen. Okay, the second family is Thymelaceae, which consists about 30 species of the Phaleria. And the phytochemicals detected from the plants mainly are triterpenes, benzophenone, flavonoid, and xanthone, and wide varieties of use in terms of to treat cancer, diabetic, and so on. So this is some of the compound that the student managed to isolate from Phaleria macrocarpa. As we can see, the structure consists of benzophenone and benzophenone glycoside, the second structure, and also the triterpenes. Okay, another triterpenes which has a different functional group bonded to the triterpene skeleton. Okay, the third compound is Gingerbraceae, which is very known in Malaysia, Indonesia, Brunei, Singapore, and Philippines, and it consists about 24 genera. And the phytochemical consists in this family mainly of phenolics, terpenoids, phenylpropanoids, diarrheheptanoids, and flavonoids. And it can be used in certain illness such as asthma, digestive disorder, and so on. So from one of the species, namely as Alpinia mutica, when we test or when we investigate on different parts of the plant, we manage to make a map that some of the compound only present in the fruits part, and some of the compound is only focused on the rhizome or the leaves part. So this can be one of the chemical marker to identify what is the part used, especially for the to proceed with the biological activity. So mainly the compound that shown here are phenolics compound, and also some are diarrheheptanoids from the rhizome parts, and anibadimer and auramide is from the leaves part. So even though it's come from the same plant, but when we isolate the compound from the different parts of the plant, we can see that the varieties of the compounds or the varieties of the class of compound depending on the part that we investigate. Okay, so we also did on the essential oils, okay, essential oils from the Alpinia species. So what we can see here, again, a different part of the plant give a different major constituents of the compounds. As we can see here, from this part, the major compound is camphor. But when we try on ripe and unripe sample, again, the composition of the major constituents are different. The major constituent is transpanisol, which is one of the sesquiterpenoid plant. And then the composition of the ripe and unripe part are different, okay, together with this data. Okay, the next plant is Rutaceae, which consists about 1,700 species in Malaysia. And the phytochemicals detected mainly are alkaloid, flavonoids, glycoside, terpenoid, and so on. And it can be used for the treatment of toothache, gout, and so on. Okay, so this is one example of the plant species. We call it as Citrus macroptera. As you can see here, different kind of plant, different kind of compound managed to be isolated from the sample. Okay, and we compared the composition of the plant from different country. We also see the varieties of compound isolated. Okay, we also did on the essential oil and the different in terms of part give different major compounds of the chemical constituents. So the focus for this talk is on the Cassia species. So this is the photo of the Cassia sanguina, which is quite similar to the Cassia alata that we have in Malaysia. So as we can see here, the biological activity consists of the antioxidant, anti-ulcer, and on the phytochemicals, it consists a lot of anthraquinones, flavonoid, tritipene, and alkaloids from different extraction methods, which are maceration and Socklet extraction. So the extraction part is very important for us to start with the isolation of the bioactive compounds. So the extraction can be divided to 3 parts, which are maceration, Socklet extraction, and ultrasonic assisted extraction, with different time that we undergo for the extraction process, 3 days, 24 to 48 hours, and for UAE it requires less time. And the extraction, as I mentioned just now, is very important because this is the first step to separate the desired natural products or desired compounds from the raw material. So it's very important to choose the right extraction method for us to get the targeted targeted bioactive compound from the plants. So to get the maximum yield for the extract and isolated compound, so we did the optimization process on the extraction using the RSM, response-based methodology. And yeah, this is the data that I get from the Web of Science, meaning that the UAE and the RSM is very important now in order to do the extraction. And then from this, we choose 3 variables, which are time, temperature, and solvent to sample ratio. So from the data, it shows that the most factor that influence the yield of the extraction are the extraction time and the extraction temperature. So from the data, we choose the best extraction time is 25 minutes, extraction temperature is 50, and solvent to sample ratio is 10 milliliter per gram. And the yield that we predicted from the optimization is 1.64%, and the actual yield that we get is 1.65%, which is very close to the predicted yield. Okay, so we compare the extraction techniques from the UAE, maceration, and Socklet, and the high extraction yield is from the UAE, which consists which consume less extraction time, which is only 25 minutes, and we managed to get 1.65% of the yield. Okay, so this is from the data on the different extraction method on bioactive compound isolated from the plant, which is no report so far on the UAE extraction method. So I just skip this one. Yeah, I think how many minutes left?
Your time's almost up.
Oh, okay. So yeah, this is all the compound from Cassia, and then this is all the technique used to finally we get the structure of the compound, especially on the spectroscopic techniques. So we did the HPLC recycling to separate the compounds from the extract, and we managed to isolate 8 pure compounds from the recycling HPLC. And next is the structure elucidation based on the NMR spectra and also the MS. Yeah, and when we're looking at the compound, it's very quite similar actually in terms of the structure. It's just that the different in terms of the substituent bonded to the benzene ring. So we want to see how good or what is the relation between the different substituent bonded to the benzene ring towards the molecular docking analysis. So we did on the antioxidant activity. So from here, it's found that the compound named 7-methyl-fission is good as antioxidant activity. And we also did on the antibacterial activity, and we found that compound number 6 shows the best antibacterial activity compared to other anthraquinones compound. So to end this presentation, to start off with biological activity, we should know what is the best optimization on the extraction process, followed by structure analysis of the bioactive compounds using the spectroscopy measurement. And then we also can synthesize the bioactive compound if the compound isolated from the plant material is in a very small amount. And finally, we can do the molecular docking to see what is the best biological activity that suit to the isolated compound. So thank you very much for your participation. Thank you.
Thank you. Thank you, Dr. Nuraza, for sharing on the on bioactive compounds, phytochemicals from cassia. I hand over back to Professor Lisitsa.
Okay, so I introduce Nikita Ionov. He is from the Institute of Biomedical Chemistry that is in Moscow, and he's working in Department of Bioinformatics on the product which is called Fit for Health, and that continue our topic on medical plants. Nikita, are you online?
Yes, hello.
Hello.
Okay. Good afternoon, dear colleagues. Before I begin, I would like to thank the organizer for the opportunity to talk about our work on the development of a database of Russian externalized medical plants, phytocomponents. Natural chemical compounds play an important role in the development of new drugs. For example, between 1991 and 2019, more than half of all drug molecules were based on natural products. Compared with synthetic molecules, natural ones have a number of advantages. The first is the presence of experience of traditional use, which allow you to get an idea of the effectiveness and safety. And second, is the natural compounds cover a large area of chemical space and have a large set of possible biological activities. Extensions of the natural products pharmacological potential is possible with the use of chemoinformatic methods. Application of in silico methods for Russian medical plants phytocomponents was complicated because of first is the absence of the data set of the extraction in suitable form from computational analysis, and second is the evidence of existing database does not characterize the phytocomponents composition of the Russian pharmacopoeia medical plants. To solve this problem, we conduct work on the development of a database Russian medical plants phytocomponents. Our data are based on the species name sets of plant used as herbal raw material presented in 14th edition of Russian Pharmacopoeia. Taxonomic characteristic of the collected plants were obtained using the NCBI taxonomy. Known synonyms of species names were collected and manually annotated using the plant list web resource. The set of synonyms was prepared taking into account their occurrence in the text of scientific publications found using the PubMed or Google Scholar. For formed set of plants, information on the representation of chemical compounds, their composition was searched on the basis of scientific publication and various natural product databases. For the compounds collected, their structures, values of physical-chemical properties, and information about their interaction with human molecular targets were extracted. Biological activity profiles were also calculated for the compounds collected using the program PASS Refine 2022. This version of the program will allow the prediction of the almost 2000 type of biological activity with average accuracy of 97%. The PASS algorithm is based on naive Bayesian classifier and the representation of structures by M&A descriptors. The results of PASS prediction is set of biological activity with two estimates. First is PA, is probability of to be active, and second SPI is probability of to be inactive. All activities with PA more than PI values are considered probable. As a result of our data mining work, we have formed a data set which includes 268 plants and information about more than 3,000 phytocomponents in their composition. The number of records describing the representation of chemical composition in the plant's parts more than 9,000. These compounds are supplemented with this information with their structures in forms like mole, inchi, canonical smiles, or inchi K, and physical-chemical properties like molecular weight, rotatable bonds count, number of aromatic rings, and other. The total number of records containing information of the results of in vitro testing for interaction with 802 different molecular targets is more than 50,000. Also, record numbers of pharmacological effect and mechanism of action for individual compounds predicted by PA more than 0.5 threshold is more than 135,000. Also, we have developed the web interface to access information in Fit4Health. On this resource, three types of search can be used to access the information. First type is a search. It's also want to use text queries in 4 categories: search by compounds, search by plants, search by target, and search by past activities. Second type of search is advanced search. It allows the user to limit the search results by specific particular parameters, including plants, molecular targets, past predicted biological activity, and range of compounds' physicochemical properties. And last type of search is search by structural similarity. Entered by structure of a chemical compounds for subsequent structural similarity search is possible using Marvin.js. The similarity search algorithm is based on the calculation of similarity metrics between the query structure and the database structures represented as MNA and Q&A descriptors. The algorithms for calculating similarity between structures represented by MNA descriptor use the Emoto coefficient, and the structures presented by Q&A descriptor, the Todeskine approach. Okay, thank you for your attention. More details about the database development process can be found in this publication. Also, Fit4Health web resource can be available by link. Yes, thank you so much.
Thank you very much for your presentation. And we continue the topic with a presentation from Professor Parokov, who is head of Department of Bioinformatics in the Institute of Biomedical Chemistry, Moscow. And I just to characterize that this department include actually 3 branches. One is the structure-based drug design. Another one is modeling of 3D structures. And the third one is supercomputer parallel calculations. Professor Parokov.
Okay, so first of all, I would like to thank the organizers for giving me an opportunity to present some results of our studies at this online roundtable on bioinformatics. And there is no need to argue that natural products are very important for drug discovery, because Nikita already presented some arguments. So I just would like to see that due to large chemical pharmacological diversity, natural products have some hidden pharmacological potential, which could be evaluated using in silico methods. When we see what in silico methods are used in drug discovery, it's necessary to cite the publication of Jorgensen in Science, who actually asked if any new drugs exist that has been discovered without computations. And currently we have both target-based drug design, which requires a 3D structure of the protein target, and of course molecular modeling for assessment of interaction between the target and ligand. Also ligand-based drug design, which is based on the structures of ligands, and some machine learning methods are applied to estimate the biological activity. In between, there is network pharmacology, which includes the reaction of different biological and pathological processes on the interaction of some targets with some ligands. Our long-term project is associated with ligand-based drug design approach. It started more than 30 years ago, and at that time, some theoretical foundation of methods has been developed, including local correspondence concept, novel structural descriptors, original bias classifier. We created high-quality training set and knowledge base of structure, mechanism, effect relationships. We started from 9,000 of biologically active compounds in the training set, and we are able to predict about 100 activities with average accuracy about 0.85. Currently, we have the training set more than 1.6 million biologically active compounds. We are able to predict more than 8,000 activities with average accuracy 0.94. And it's necessary to stress that in 1999, we introduced the first in the world software for prediction of bioactivity spectra in the internet. And it's still developed, and currently it's used by more than 40,000 users from 90 countries, and millions predictions and 1,000 publications were developed based on such predictions. Biological activity spectra actually include the following categories. It's pharmacotherapeutic effects, mechanisms of action, specific toxicity, interaction with some anti-targets, metabolic terms, influence on gene expression, and action on transporters. On this slide, you can see the example of prediction for well-known phytochemical quercetin. Actually, you can see that antioxidant activity is predicted with high probability, and its probability to be active, probability to be inactive is quite small, and the list of activities is quite large because this structure has similarity to many different biologically active compounds. There is no some particular features which discriminate this structure from many others. To analyze the past predictions, Alexey Lagunin have developed a program called PharmaExpert, which actually allowed to use past predictions as the input file, then visualize the relationships between effects and mechanisms, then actually try to find some activities, combination of activities with some libraries of compounds, etc. Also, PharmaExpert allowed to analyze drug-drug interactions. Here you can see the interaction of two drugs, but in phytoconstituents, sometimes we have many different phytocomponents which also could be analyzed for their additive or synergistic action. Thus, using FAS and PharmaExpert, one can identify the putative mechanism of action for natural products that are used in folk medicine, determine the screens that are most relevant for a particular class of natural compounds, select new hits with given biological activity profile among the natural products available in-house or in commercial databases, reveal new effects and mechanism of action for known natural products, and find different natural products combinations with synergistic or additive pharmacological effects. I give you just two examples. More examples actually were listed in my abstract and appropriate publications. The first example is discovery of antidepressant action of Achiranthes aspera. As you can see, Achiranthes aspera is used in traditional medicine for different indications except antidepressant. And jointly with Professor Rajesh Goyal from Punjab University, India, we performed the prediction of biological activity spectra for phytocomponents of Achiranthes aspera and found that it has several antidepressant mechanisms of action. Then this result was confirmed in vivo experiment in experimental animals, and paper was published in Saran Q Environmental Research. The next example is our joint project with Olga Bocharova. She is professor in Blokhin Oncological Center. So they developed very complex composition called multi-phytoadaptogen. It includes extracts of 40 medicinal plants and about 70 major phyto components. It has many different biological activity which were already shown in the experiment. But using PAS and pharma expert, Nikita Ionov was able to actually understand the mechanisms of action, anti-tumor action, and predict some additional indications that could be the further studies. To provide more opportunities to use past predictions for natural products, we actually incorporated the results of past predictions in several databases. Phyto for Health was already shown by Nikita. Also in Brazilian medicinal plants, it's systematics database, and also in coconut, which is actually combination of many different databases or many different sources, more than 400,000 compounds. Using PAS online, many publications with past predictions of some particular biological activity which were confirmed by the experiment were published. Thus, coming to summary, I would like to say that natural products are the promising source of new pharmaceutical agents. Computational analysis based on PAS and pharma expert estimations allows to identify a hidden pharmacological potential of separate natural products and their combinations, to provide the international scientific community with the information about potential biological activity of natural products. PAS results were incorporated into systematics, coconut, and Phyto for Health database. And PAS Online is widely used by scientific community to estimate the probable biological activity of natural compounds. With that, I would like to thank you for your attention, acknowledge my current team, former team members, and some of our collaborators who took part in this long-term project. And of course, I would like to thank the different funding agencies and some companies, pharma companies that actually buy the licenses on our software or use them in their application studies. And also I will use this opportunity to introduce a 29th Symposium on Bioinformatics and Computer-Aided Drug Discovery, which will be held virtually in September 2023. And it's open for registration and submitting abstracts. Thank you once again.
Okay, thank you very much, Professor Paroykov, for representing the condensed representation of the results, which I know you invested many tens of years. And if no one mind, we will ask organizers to distribute the information about the symposium on bioinformatics among the participants of the workshop to make it like a hyperlink available for registration. So, Professor Rai, can you continue our program?
Okay, thank you. So I invite the next speaker, Dr. Sudat Lam. Dr. Lam is actually from the Department of Applied Physics, University Kebangsaan Malaysia. You have the floor.
So I'm Sudat. As mentioned by Firdaus, I'm actually based in the National University of Malaysia. First, I want to thank the organizer for giving me a chance to share the research I've been doing. Today, I decided to go a bit different, but talk about one type of splicing called mutually exclusive exon. For those of you that don't know what splicing basically is a process that expands the diversity of proteins in multicellular organism. It refers to the rearrangement and assembly of all the exons and then ultimately produce different type of mRNA and different proteins. If you're still confused by what I say, basically this is the gene, and this gene actually have 5 of the different exons. So in some of the cases, in one of the isoform, all 5 of the exon, so they are potentially protein coding, they have been spliced inside, and then after that they will produce these protein. However, in a different isoform, the exon number 3 is not included. By not having this actual isoform, the protein that produced will be different, and this will have some kind of effect on the protein functions. Currently, there's a few types of splicing. The one I'm going to focus on is actually called mutually exclusive exon. It's characterized by the rearrangement of homologous exon, where one of the exon will be retained and the other one will be spliced off. In short, in one of the isoform, you'll find the red exon, and the other one you'll find the blue exon. Although this type of splicing actually accounts for less than 5% of them. Alternate splicing has been implicated in diseases like Alzheimer's and also in cancer. So far, alternate splicing is well documented at the transcript level. However, when we go to proteomic experiments, we actually found low evidence of that. Why? Because some of this isoform maybe happens in very small quantities, or some of them will be happens only at specific conditions, tissues, developmental stages. These making finding them using proteomic experiments a bit hard. However, all of those isoforms that detected from the proteomic experiment, they are significantly enriched by mutually exclusive exon, and that's why we are focusing on that. Currently, there's some small-scale study that are actually looking on that. So they found that this mutually exclusive exon usually doesn't change the protein structure much. The difference you can see is just changing of a few residues. However, in this specific example, the variable residues is actually shown in purple and also the blue on the right. And this example, we can see that these residues actually lies very close to an AT binding site, meaning maybe tuning the protein's functions a bit in different isoforms. Currently, there's some studies showing that MXC is evolutionary conserved. The study suggests that MXC actually had an ancient origins up to 425 million years ago. That's so long. Just to say, so currently there's no large-scale studies on MXE in whole genome, and we decided to use structure analysis tool developed by our group to analyze the mutually exclusive exon in 5 organisms. To get the data, initially we went and searched for a database and we found one called Cassiopeia. However, this Cassiopeia actually contained only the fly MXE. But for the other organism, we need to predict them using the sequence we obtained from the ensemble. So in the end, we got these many of MXE. There's about thousands of them, corresponding to about 7,000 genes, and they are coming from 2 different types of fish, mouse, human, and also fly. First question that we ask is, what kind of function do all of these MXE genes actually have? So we use a software called David, quite easy to use. Just put in the gene and you get some kind of find out the significant and reach gene functions. And we found that these MXE genes tend to be membrane protein. Interesting. And then the other one, they are also enzymes such as kinase, transferase, or even oxidoreductase. Because we are a structural group, so we try to provide and see whether can we annotate them with structural informations. And we managed to cover about 65% of the MXE with some structural information. Just to recap, usually these MXE pairs doesn't have a lot of changes in the structures. What they are happening is just having some variable residues changing here and there. Then the next question where we ask is, using our data set, how often do we see these variable residues between the MXE pairs? We can see less than 10, usually they have less than 10 variable residues with a median of 6 residues, meaning the MXE residues that's variable are actually not a lot. Then we started asking questions that try to point, can we get some kind of functions to it? The first question we asked is whether these variable residues are actually exposed to the servant. So we first compared the MXE region versus a random region, basically from the same protein. And we can see that overall, the data seem to shift to the right, saying that these MXE regions are way more exposed compared to a random region in mouse. And the same happened for the other organism. It's also significantly a difference with them. We did a test with the tests. The next question we asked is, within the MXE region, how about the variable residues versus the non-variable residues? We do see similar patterns. We see that variable residues seems to be more exposed compared to the non-variable residues. This telling us all of these residues seems to be more exposed, so they are not going to affect the protein structure, the folding of them, but more likely to tune the function of them by those that are going to like interact with protein interactions or those that interact with a ligand and stuff. Next, the question we ask is, you have all of these variable residues, do they actually have a big physiochemical changes? And we use the McLachlan score to test the physiochemical properties of that. Keep the story short, we actually found most of our variable residues actually have large physiochemical changes. Interesting. So there's not many residues, but most of the residues are actually exposed and they tend to have large physical chemical properties, the changes of them. The next question we ask is whether these variable residues are actually close to any functional site. When we mean functional site, we're talking about the protein-protein interaction site, catalytic site, ligand binding site, allosteric site, and also our predicted functional site where we predict them using evolutionary conserved information. Because there's some of the cases, there's a lot of variable residues, we cluster them, produce variable region clusters, and we do that because we want to reduce the noise that we get from our data. Now we can ask our questions, how often do these variable region clusters lie close to functional site? We did compare our result to a random model as well, and this is the result we get. For CSA, we don't the catalytic site, we don't really see a strong result there, but we do see some interesting result from the protein-protein interaction site, ligand binding site, our predicted functional site, and also allosteric. Again, so this is telling us these residues are close to this kind of functional residues, probably having more chances to tune the function of it. That's interesting. And now we're going to continue and look at two interesting examples here. First one is actually an enzyme called dehydropyrimidase. This is actually found in the fly, and this enzyme is actually implicated in biological process such as the Notch signaling. And then this zinc ion basically serves as a cofactor of this enzyme. So we can see that in one of the isoform, this histidine is actually there to interact with this zinc ion. However, in another isoform where the variable residues actually change to the residues actually be changed to glycine now. Glycine and the side chain doesn't really come out, so it cannot interact with zinc molecule. What will happen to this protein? We speculate this may changes they may weaken the ability of this protein to really bind to this zinc ion as such as a cofactor. Another example, we look at a protein kinase where it's basically a part of the cycle that we all know. When we found these variable residues are close to the protein-protein interaction site and also the allosteric site. And another interesting thing is what we found is we actually found that there's a lot of cancer mutated residues actually hitting the same location in this specific example. Then we started asking questions whether these cancer mutation residues actually coincide with MXE variable residues and also with the functional residues. And we found that 35 out of the 40 MXE regions was significantly closer to cancer mutation and also functional residues. This suggests cancer may dysregulate the same functional region as the MXE events. Another thing that we want to mention, MXE regions, because they actually the membrane protein and also surface exposed, we think there are promising druggable targets. One thing that's cool, because if we can target a minor MXE splice variant, it could be very advantageous because the expressions will be more tissue specific and it will not jeopardize the general functions of another isoform. In conclusion, we found that MXE event, the variable residues, doesn't really change the stable folding of the proteins, but rather tune the function proteins a bit. The same kind of protocol that the analysis has been applied to other kind of studies. I'm so sorry that because what COVID hit us, we started looking at the SARS-CoV-2 receptor binding domain versus the ACE2. We also look at the N-terminal domain of spike protein and their interactions with sugar. And currently, we're actually looking at variants of the humans interacting with SARS-CoV-2 and also mutation that may be associated with certain plant trait. Basically, I want to thank my collaborator on the splicing paper, Christina Rangel, Jonathan Lees, and also Madan Babu. I also want to thank my mentor in the National University of Malaysia, Prof. Firdaus, and also Prof. Wan Q. Lien, and my funding agents, basically our university and also the Ministry of Higher Education of Malaysia. And you all for listening to the call. Thank you so much.
Thank you, Dr. Lam, for sharing that regarding the medical implications of the mutually exclusive exons. Can move on to the next speaker, Professor Lisitsa.
Okay. So I introduce two speakers, Arina Gardieva and Anastasia Valueva. She is young research engineers working in the Institute of Biomedical Chemistry, and they work in the Laboratory of Nanotechnology, and they will present shortly information about how we deal with omics technologies and how do we use atomic force microscopy in high precision research. Please, who will? Oh, Anastasia, you will start. Okay.
Hello, dear colleagues. Yes, I start. And today I would like to tell you about two topics, about an arsenal of ABMC and also about detection using AFM. Let's begin. The global mission of ABMC is development of methods for creating a molecular picture of healthy and ill persons. There are 4 directions in our institute: post-genomics technologies, drug delivery using nanoparticles, bioinformatics, and also single molecule science. And about single molecule science, I would like to tell in more details because it's a new approach which could help expand our knowledge about biosystems and biomolecules. And what's about single molecule science? We need some ideas from Avogadro, and we can calculate how many molecules in solution with different concentrations. And we could see that in nanomolar concentration, in 1 microliter volume, we have billion molecules. But if we have 10 to negative power of 18 concentration, we get only 1 molecule there. This calculation is important when we need to predict disease in early stage. And we need more sensitive method for the detections. And for this, we must to scale sensor element of our devices to nanometer units, to sizes of nanomolecules. And this approach is realized in molecular detector, which could help us to receive signal from 1 molecule, like on this joke picture, where molecular detector help determine a suspicious object among average same objects. And all these ideas inspired our researchers to create unique scientific facility, Avogadro. There are several types of molecular detectors in Avogadro facility. For example, nanovar detector for studying proteins and nucleic acids, nanopore detector for nucleic acids, and AFM detector. Also, our facility with mass spectrometer and MOL database, where we store our results from our devices. And all of these help us to understand molecular portrait of person. And I want to focus on AFM detector and mass spectrometer in later. AFM detector has sensor element, which called cantilever. Also, it looks like a sharp, small tip. And AFM scanning associate with maybe blind person who take information about objects when he touch for it. For example, AFM allows visualizate and calculate biomolecules like this: 1 sheet, 2 sheet, 3 sheet. Also, it allows manipulate with biomolecules like this. We can modify our cantilever with something compounds and look response from biosystem. And also, it allows determine properties and activity of single biomolecules like this. This is lazy worker, this is hard worker, for example, in enzyme population. And what we need for successful AFM scanning, we need surface like fish rod. Also, we need some surface modification we must do surface more attractive for biomolecules. And also we need volume of solution, maybe protein solution or solution of serum or plasma. All of these called fission method. Fission is in formula looks like this. It leads us from 10 nanomolar concentration in solution to millimolar concentration on surface with factor of concentration 10 for power of to the power of 8. And fission could be biospecific when we know which protein we want to detect. In this case, surface is modified by antibodies and aptamers. And also fission could be chemical when we want to take proteomic inventory of the sample, when we want to take most of molecules from our sample. In this case, we use cross-linkers as special chemical compounds which can make bond with surface and with biomolecule. Also, chemical fission we can use in this case when we need for antibodies immobilization on surface. And then we go to our attempts for coaching BCA protein using AFM chips. Purpose of the study choose the condition for successful detection and identification of the protein from the AFM chip. We use uptest marker with terminal amino groups. For modification, we use photo cross-linker based on benzophenone. And also we use 2 different concentration of solution for our experiment. There is scheme of our experiment. We incubate our chip in BSA solution with ultraviolet radiation. Then we wash our chip, dry our chip, and scan with AFM. And what we get, there is AFM images and cross sections. And in the case of sub-micromolar BSA solution, we can see lower fragments on every AFM scan, and height of these objects is about 10 nanometers. In the case of nanomolar BSA solution, we also could see layers on the surface, but its height is smaller, about 2 nanometers. And also we have control experiments, of course, and we change one parameter in every control. For control 1, we used for incubation protein-free solution. For control 2, we used AFM chip without modification by photo cross-linker. And for control 3, we didn't use UV radiation. And we can see there are no lower fragments on these pictures. And also the number of non-specific objects with height more than 1 nanometer doesn't exist the noise signal on these pictures. And summarizing all my words, we utilize BSA molecules from different solution from layers on the modified AFM chip. Heights of layers are 3 nanometers and 2 nanometers respectively. And there are no layers in our control experiment, and we think that photo cross-linker give high hopes in the field of research of concentrate solution. And next, I would like to give words to my colleague, Arina Gordeeva, about MS part of this research. And also I want to tell thanks IBFC team of especially Laboratory of Nanobiotechnology team, and I appreciate for our teamwork and opportunity to take part in interesting and vital research projects. Thank you for your attention.
Thank you very much for your report and very nice sheets Please, we will continue with a report from Arina Gardieva with the topic of atomic force microscopy. And please, Arina, can you switch on a camera if you do have one, not only presentation, but also a camera to see you?
Okay, good day, my dear colleagues. Today I present to you my report, mass spectrometry identification, the BCA covalent protein immobilized on the chip for atomic first surface. Today I will give you IFM MS part. Secondly, we discuss MS part of our experiment. After that, we talk about results of experiment, and of course, we talk about future prospects. Firstly, I will give information about IFM MS approach. As we've seen, the results very dependent on the crosslinker, and today I will give information about two types of crosslinker. It's IFM chip for detection in mass spectrometer. Firstly, I will give a scheme of our big experiment. Firstly, it's a modification and activation of the IFM substrate. Secondly, it's a covalent protein immobilization. Thirdly, IFM scanning. After that, we preparation IFM chip for the mass spectrometry measurements, and of course mass spectrometry measurements and MS data processing. It's a bioinformatics part. MS approach, it's a really efficient approach, but the lower sensitivity than instead we develop this method for entire all arsenal of the MS to the MS approach. Important to say that large of number of steps can lead to sample loss or contamination, unfortunately. So it's necessary to select sufficient sample preparation for our experiment. Firstly, it's a BCA covalent protein immobilized. After that, it's optional steps, alkylation and reduction. Thirdly, it's a necessary step, proteolysis, as we know. After that, dissolving, it's an optional step as concentration method. And of course, mass spectrometry identification, as I said, and database search, as you can see, different bases. So we try alkylation reduction, concentration method, and dissolving. And we use two types of crosslinker. It's a DSP crosslinker and SAGBB crosslinker. It's a IFM-MS approach. So, two types of crosslinker on a mica surface. Two types of experiment, alkylation and reduction, and without these steps. After that, of course, proteolysis with trypsin and mass spectrometry identification and data processing. Can we discuss our results of MS analysis? Firstly, it's a DSP crosslinker. Secondly, SAGBB crosslinker, solution, and of course, free controls. As you can see, DSP crosslinker have less unique peptides than SAGBB crosslinker. The maximum number of undefined peptides in our experiment is SAGBB crosslinker. As you can see, it's a SAGBB crosslinker and less DSP crosslinker. Of course, we've done protein solution, and also we've done nanomolar SAGBB crosslinker. It's also interesting result for us. Let's go to a visualization. It's a Venn diagram. Firstly, it's a solution. It's a violet, green DSP crosslinker, and SAGBB, it's pink color. As you can see, SAGBB crosslinker has 7 unique peptides. DSP crosslinker unfortunately has 0 peptides, and solution, it's a 4 peptide. So SAGBB crosslinker more interesting and sufficient for our experiment. As you can see, visualization of the BCA peptides registered in mass spectrometer analysis. SAGBB crosslinker, it's a red color, DSP crosslinker green color, and another with alkylation and reduction. So SAGBB crosslinker has a lot of colored in red in our experiment. Instead, DSP crosslinker, as I say. So, of course, we checking our results in MSMS method. As you can see, 2 peptides from our experiment from mica surface, and it's one of the biggest peptides. It's a good result for MSMS spectrum. Let's go to results. SAGBB crosslinker has been more efficient, more peptides are defined in proteolytic mixture on the surface, and protein immobilized on the IFM chip via SAC-BB open on the surface in such a way that more free sites for trypsin available. In conclusion, I want to say that these results can be found application for the detection of biomolecules and in the development highly sensitive diagnostic system based on microarrays when immobilized, we can use it, this type of crosslinker, especially SAC-BB. What about future prospects? Of course, it's the aptamers antibody crosslinks microRNA. As you can see, AVA works in this way, so it's a protein inventory task. It can be plasma, can be model protein, it can be all with crosslinkers and aptamers antibody and etc. So thank you very much for ABMC team, Laboratory of Nanobiotechnology. A lot of words of thanks for support and idea and over time working with us. Thank you for your attention.
Irina, thank you very much for your report. And we don't use questions, but
Okay.
we type questions in a chat. And so you have seen from these two reports that we are doing, first of all, a mass spectrometry analysis, which include MALDI, which is matrix-assisted type of ionization, but also mainly we work with electrospray ionization coupled to liquid chromatography. And what is new, generally new in the expertise of our institute, that we combine the molecular detectors, one of which is atomic force microscopy, with further, which is a detector, but we combine it with further identification of the macromolecules by using mass spectrometry. That is a combined type of the technology. We do the same stuff for the nanowire detector, and also we do have an expertise in analyzing of genome and transcriptome by using Oxford nanopore, and that is a kind of a couple of omics technologies. Also, it is combined with cell technologies, cell sorting, and with biosensor techniques. And certainly bioinformatics, which has been presented very shortly by Professor Parokov. That is a general view of the competence of our institute. And Professor Rai, I give a word to you to introduce next two speakers from UTM.
Thank you, Professor Lisitza. For the next speaker, I'd like to call upon Dr. Muhammad Firdaus Abdul Wahab from University Technology Malaysia. Dr. Firdaus, you have the floor.
Thank you, Prof. Firdaus. My namesake from UKM. We share the same name actually, but you are obviously more famous. I hope you can hear me well. I can see my slides. So if I can steer your attention away a little bit from the biomedical theme that we've had so far into environmental and biotechnology, but actually the omics methods and the pipelines that are used in biomedical or environmental are more or less similar. So there's something to be shared among these two fields. So I like this diagram from DeHaan et al. 2021 when they were describing the use of microbiomes from natural resources and how do we exploit these microbiomes to achieve all the different sustainable development goals. So my lab is interested in looking into sustainable bioeconomy, a little bit of climate change mitigation by looking at the waste treatment, and probably delving into healthy people, animals, and environment domain by working on the bioremediation aspects. Our interest is basically to use the microbiomes, exploit them, if I can use that word, and achieve the impact that we want to make human lives better. The focus is on bioenergy production because, as we all know, microbes are versatile and can be used to generate renewable energy fuels from the various types of biomass and biological waste because of their high metabolic diversity. The interesting thing is that the metabolisms can be rewired by either acclimatizing the microbial community according to the different physical chemical parameters, or we can metabolically engineer them, or even community engineering can also be done as well. So the different biofuels can be produced by different microbes and the microbial community as a whole, each of the members of the community performing different function. So in this figure here, you can see that from different types of substrates through the different metabolic routes, we can produce all sorts of bioenergy related fuels. So what is the kind of substrate that should we investigate? So as we all know, Malaysia is one of the biggest producers and exporters of palm oil and palm oil products. So you can see here the proportion of palm oil in the global oil and fats production is quite significant from 2016 to 2020. And on the top right here, we can see that there is an exponential growth in oil and fats consumption. However, the production of oil and fats are expected to be linearly growing. So there is going to be a high demand for oil and fats beyond 2050, making palm oil production very, very important. Actually, the process of producing palm oil from oil palm actually generates many different types of waste. So through the different methods employed in the mills. So it can be categorized as two types. One is solid waste and another one is liquid waste. So the liquid waste is called the palm oil mill effluent. And it is actually very highly polluting, but the good thing is that it is rich in organic matter that can be used by the microbes to convert into various products that we want. So one commercially applied use is the conversion of palm into biogas, which is particularly biomethane. This is one example of the biogas digester using covered lagoon method. So the community here has been acclimatized to produce methane. But the good thing is that we can actually use the same community by acclimatizing it to produce hydrogen. So what is good about hydrogen is that it is a clean energy carrier. It has high energy yield. And if you burn hydrogen, you only get water. There are several routes for the production of hydrogen using biological methods. And my lab is focusing on the dark process, which is also called dark fermentation. But there are still major challenges faced by this method, which is low biohydrogen yield and the practical conversion, which is only about 30% of the theoretical conversion, which is like 1 mole of glucose producing 12 mole of hydrogen. We have not yet reached this stage. So research is still needed to optimize the microbial community for the conversion. And on the right here is just the different stages involved in that hydrogen. And if we did not stop the conversion process, it is going to be converted into methane and carbon dioxide. So these are the different stages again. And this list just shows the different taxa associated with the different stages and the different key enzymes involved in the different stages. So what we were doing in the lab was to acclimatize the microbial community from anaerobic digester to produce hydrogen. So what we are doing is to increase the palm concentration that we are exposed to the microbial community from 10% to all the way to 100%. And you can still see here there is production of hydrogen is quite lower. And what we did was to take samples along the way and observe the microbial community changes using amplicon sequencing. So you can see here that there are significant changes in the archaea population in blue here as we increase the palm concentration to 30%, and at 100% the archaea is significantly suppressed. So the diversity of the microbial community also changes from the untreated or raw anaerobic sludge, and when we expose them to palm, they decrease in terms of diversity. So you can see here the dominant community at 100% palm concentration is from the Firmicutes phyla. So I'm using both new and old phyla names here because old habits die hard, I suppose. So, and then we tested the different pretreatment temperature on the inoculum performance to see which temperature that favors the highest hydrogen producer. And we found that at 65 degrees, we got the highest hydrogen production using the same inoculum as before. And this time we managed to do some shotgun metagenomics to observe the microbial community changes and also the functional changes as well as happening after and before the pre-treatment. So we can see here that before treatment here, community are more or less similar, but when we incubate them, they are more or less diverge into different community structure. And as before, pre-treated sample give rise to significant enrichment of Firmicutes members here, as you can see at 50 degrees, 65, 80, and 100. And raw sludge has more diverse community. So this is actually the total taxa composition from a different treatment and also the raw sample group into one, just to show you that we have high dominance of Firmicutes and Proteobacteria as well here. And we detected some urea archaeota from the archaea community, mostly from the raw untreated sludge sample. We dig deeper into the data set and found that true enough, it was the Firmicutes. The orange one is the enriched taxa. So Firmicutes is highly enriched after fermentation. And there are also a few other taxa that are suppressed after fermentation. Of course, the archaea is suppressed and a few other members of the microbial community. So here it shows that the 65 degrees pre-treatment enriches certain Clostridium taxa, while in the raw untreated sample, you have Candidatus, which is commonly found in an anaerobic digester, a few archaea, and also some proteobacteria as well. So we also analyzed the potential functional changes before and after fermentation. And this is actually according to the seed classification of functions. So these are the relevant and also related functions that we are interested in. And as we dig deeper into the cellular processes function, if we zoom up at the prokaryotic cell type differentiation, we can see there is an increase in sporulation, which is associated with hydrogen production. And then when we look at carbohydrates function, we can see that there are a few functions affiliated with sugar fermentation is upregulated here. And the more interesting one is the fermentation function is upregulated as expected. And methanogenesis, which is conversion of hydrogen into methane, is downregulated in the community. And finally, we look at the fermentation function, the next level, and we found that the upregulated function is associated with the mixed acid fermentation and the glycerol fermentation, while acetolactate synthase subunits is downregulated, which is unwanted in hydrogen production. So we also assembled some MAGs or metagenome assembled genome from the raw sludge sample, and we found 36 high-quality MAGs and 57 medium-quality MAGs. And the figure here shows the distribution according to the different phyla. And we can see here 2 archaea or maybe 3 archaea types are highly abundant in the MAG assembly, which is Methanobacterium, Methanothrix, and also Methanolinia. So these are currently being analyzed in order to know their methanogenesis pathway. So we also assembled several bacterial MAGs, a lot more complex than archaea MAGs, probably. And at the same time, we look at the correlation between archaea and bacteria in the anaerobic sludge sample. So this is still work in progress. I hope to describe and explain to you a little bit more once all the analysis has been done. So finally, I would like to acknowledge all our students, members of the lab, past and present, our industry and university collaborators or funders. And this is the last slide. I would like to draw your attention to the training course that I'm organizing in Johor Bahru in August. So this is funded by ICGEB. The last time I checked, Russia is part of ICGEB. So if you know any colleagues or students interested, spread the news. Thank you very much. And back to you, Prof. Firdaus.
Thank you, Dr. Firdaus.
Sorry, Professor Firdaus, can I just comment that it makes sense to send that to organizers to spread the information automatically into our community. That is very interesting course, and I think many of my students will take it.
Sure, thank you.
Should post that to the organizers, Dr. Firdaus. All right, thank you, Dr. Firdaus. And I'm going to call on the next speaker, Dr. Shazwani Itri Amran. also from University of Technology Malaysia. Please, you have the floor.
Yes, thank you, Prof. Firdaus. Thank you to the chairman and also the organizer for having me. So today I'll be sharing with you about my research related to mutation-induced structural instability of G6PD variants. So I will start off my presentation with some information on G6PD deficiency. So G6PD deficiency is a common enzymopathy characterized by decreased level of enzyme activity leading to destruction of the red blood cell. Even though G6PD present in most type of the cell, but G6PD deficiency is usually associated with hemolytic anemia. So in normal red blood cell, G6PD enzyme is responsible in generating the NADPH, and this NADPH in turn plays an important role in redox metabolism in the red blood cell by generating reduced glutathione from the oxidized state. So this reduced glutathione acts as a major endogenous antioxidant that protects cells against oxidative damage and from free radicals such as the H2O, where we convert this hydrogen peroxide to water molecule, which is harmless to the cell, and therefore we can avoid oxidative damage. However, mutation in the G6PD enzyme will result in reduced enzyme activity, therefore reduce NADPH level, and decrease the level of reduced glutathione, and in results, it will increase the oxidative damage and results in hemolytic anemia. And the reason why we are interested in this field because the prevalence of G6PD deficiency in Southeast Asia, Middle East, African region, and also part of the Europe is quite high. And this disorder has been associated with malaria infection, cancer, and recently COVID infection. So in the past 36 years or so, over 230 genetic variants have been identified, most of which are single base substitution leading to amino acid replacement. And the mutation result in protein variants with different levels of enzyme activity that are associated with a wide range of biochemical and also clinical phenotypes. And according to the level of enzyme activity given on the slide, the World Health Organization, WHO, has classified variants of G6PD into 5 classes, class 1 until class 5. Where class 1, which is a severe deficiency with chronic hemolytic anemia, usually represented with no detectable or less than 1% of enzyme activity. Whereas class 2, which is also a severe deficiency, represented by acute hemolytic anemia, having less than 10% of normal enzyme activity. Class 3, having a moderate deficiency with having 10 to 60% enzyme activity, whereas class 4 and 5 are asymptomatic with more than 60% normal enzyme activity. So the G6PD enzyme structure is relatively very big. It can present as homodimer or tetramer in its active state. So on the slide, we can see the homodimer structure made up from 2 monomers in green and also in cyan. So in each monomer, we have 1 glucose 6-phosphate substrate bind to the monomer and 2 molecules of NADP+. So one of the NADP+ molecule involved in enzyme catalysis, whereas the other NADP+, which is located at the dimerization site, is responsible for enzyme stability. So if you can see the other labelings here, represented the name of the G6PD variants represented by different geographical names such as Plymouth, Taipei, Coimbra, Yucatan, Andalus, Mexico City, Nashville, Valladolid, Union, Wisconsin, Durham, and so on. So this mutation, regardless of their classes, whether they are class 1, 2, or 3, they are scattered all over the enzyme structure that adversely affected the enzyme activity. So looking at this table here, it is a compilation of kinetics data for different G6PD variants of different classes. It is noteworthy that variants with mutations located in different regions of the structure showed lower catalytic activity given by the KCAT value, which is the wild type is 230, whereas the variants are lower than the wild type value. And based on this kinetic data also, we can't really tell if there is a specific pattern or range for class 1, 2, and 3. Plus, not all mutations affect the affinity of the G6P substrate and also the NADP plus cofactors. For example, if you can see here, the Yucatan variant, which belong to class 1, has somewhat decreased catalytic activity. However, the affinity towards the substrate and the cofactors are similar to the wild type. So we hypothesize that reduced enzyme activity does not only caused by changes in the affinity of the substrate and the cofactors, but it could also be associated with impairment of protein folding or altered dimerization. So we also understand in cellular setting that G6PD enzyme exists in dynamic equilibrium of the dimer and the tetramer in its active state. So the equilibrium is highly influenced by the ligand, which are the G6P substrate and also the NADP plus cofactors. So increased G6P concentration will dissociate the dimer into its inactive monomers, while increase the concentration of NADP plus will shift the equilibrium towards the tetramer. So what we did, we examined the structural changes using molecular dynamic simulation, and we simulate the dimeric G6PD enzyme structure using various trajectories and computational analysis. We collect all the data and draw out some conclusion from it. So this is the list of variants that we have covered so far, representing each class 1, 2, and 3. The aim is to find out the similarities and differences among different classes of variants. So we focus on several important regions on the enzyme structure. As you can see here, the first important region that we focus on is the substrate and NADP, catalytic NADP binding site, which is governed by this beta E alpha E loop. And on this loop, we have the catalytic amino acid, which is lysine at position 171, involved in the catalysis of the enzyme activity. And then next, we also focus on the structural NADP binding site, which spans both in the monomeric subunits in the dimer. Then we also focus on the dimer interface and also the tetramer interface. So through our detailed analysis at the dimer and tetramer interface, we found that the dimer interface is stabilized by 2 salt bridges and 1 hydrogen bond. So these 2 salt bridges link lysine and glutamate together in the alpha I helix, and also the hydrogen bonds connecting the 2 aspartate from the 2 monomers connecting the beta and sheet. And in the tetramer interface, we found that the tetramer is stabilized by 2 salt bridges in the alpha J helix connecting the lysine 275 and glutamate 347, and also lysine 290 with glutamate 287. To summarize our finding, we found all class 1 variants exhibited similar structural changes at the dimer and the tetramer interfaces. So in this slide, we present a few class 1 variants given by Shinagawa, Bangkok, and Guadalajara of class 1. So we found that Shinagawa and Bangkok shows the similar alteration at the alpha 1 helix and the beta N region, and at the dimer interface, but less structural difference at the tetramer interface. However, interestingly, in Guadalajara, the interaction in the beta N region is conserved. However, we find dissimilarity at the tetramer interface, and that disrupt the tetramer formation. So let's have a look at the class 2 variants. So it can be inferred that all class 2 variants exhibited similar structural changes at the alpha 1 helix of the dimer interface, and this has become the reasoning for its unstable dimer and justify their inability to establish hydrogen bonds between the beta and sheet of the monomeric subunit. However, for Vanuatu lava, we see that there is an addition disruption at the structure in the tetramer interface, and therefore loss of activity in Vanua lava variant is caused by impact tetramerization. So we are certain that disruption of the enzyme dimerization is the major contributor to loss of enzyme function in class 1 and class 2 G6PD variants. Therefore, our ongoing project is to develop novel G6PD activator or agonist that can restore the enzyme activity by stabilizing the dimer. So what we have here is the preliminary computational analysis using a molecule called AG1. So this AG1 molecule is the first G6PD activator reported to date, developed by researchers from Stanford University. So the RMSD plot showed the binding of the agonist at the dimer site could stabilize the structure of class 1 and class 2 G6PD variants. And if you can see here, in the absence of AG1, the RMSD plot for the variants is a little bit high compared to the wild type, which is given in black. And when AG1 is bound to the variant structure, the RMSD plot is reduced, the RMSD value is reduced and almost coincide with the wild type. So this serves as a basis for designing novel G6PD activator or agonist. So to end my presentation, I would like to acknowledge the sponsors from Ministry of Health, Saudi Arabia, our Ministry of Higher Education and UTM for the funding, for the financial support and facilities. And also I would like to thank to the students and collaborators that involved in this project. So thank you very much.
Thank you, Dr. Shazwani. I hand it over back to you, Professor Lijekta.
Yes, thank you. And we move forward. And the next speaker will be Елизавета Sarygina. She's an instructor working in the Center for Practical Education. That center is a part of the Institute of Biomedical Chemistry in Moscow, and its target is to proactively work with students and PhD students in the field of metabolomics, metagenomics, and bioinformatics.
Dear colleagues, thanks for inviting me to the workshop.
Елизавета,
I apologize that I can't turn the camera.
You cannot turn on. Okay, no problem.
Today, I will talk about how we can measure the accuracy of the algorithms. So I will report how simulation of next generation sequencing can be used for that. Transcriptomics is a large class of methods based on next generation sequencing. RNA sequencing has become widely used in biology and medicine because it provides highly accurate measurements for each nucleotide in a short time. There are several sequencing platforms. Illumina technology is second generation sequencing and provides read lengths of 150 to 300 nucleotides and generate hundreds of millions of reads. Their fluorescent tags are used to detect of nucleotides. Oxford Nanopore technology sequencing is based on the principle of passing RNA molecule through a tiny protein pore. Measures the change in current RNA strand move through the pore. One of the main advantages of this technology is the ability to generate long reads of thousands of nucleotides. Sequencers, Illumina and ONT, provide the raw FASTQ data format or FAST5. Computer analysis of RNA sequencing data is a pipeline of bioinformatics programs. It starts with quality control and streaming of adapter sequences if the data required it. And the next step is the alignment of the reads to the reference genome or transcriptome. Quantification of expression levels for each gene follows. Alignment is an important step in the analysis. The main purpose of alignment is to match the read sequence to the reference. This task is computationally complex due to the exon-intron architecture of eukaryotes, and various errors can occur. For example, an ambiguous alignment of reads to multiple location or fragmentation of single read into different genes. Read may contain insertions and deletions. Such data may be unmapped and lost, or mapped non-functional analogs of structured genes. These errors can lead to incorrect estimates of gene expression. This study focuses on the problem of accuracy of alignment algorithm. We used simulated transcriptomic data. It is artificial sequencing data. An advantages of using simulation is that it's a software process. Therefore, all parameters can be monitored and controlled. Two types of experimental data were used in the study. RNA sequencing of the HepG2 hepatoblastoma cell line on the MinION, Oxford Nanopore, and Illumine NanoVasec platforms. There were three main steps to the study design. The first is simulate the data using the software packages RSCM and TransNanoSim. The second is processing simulated data with alignment tools, counting reads using the HTSEQ and subreads feature count. The last step is comparison of the result of the alignment with the parameters of the simulation phase. Simulators should consider read length distribution, systematic errors and insertions, and other factors, and build a data model. It is important that the simulation doesn't oversimplify data. The basis of generating artificial data are the scoring values of the real data, HepG2. Paired and or long simulated FASTQ reads were aligned to the reference human genome and transcriptome in accordance with the scheme on the slide. When tested, GraphMap2 and Dissolved were rejected due to extremely low speed and high computational resource required. When testing mapping tools, the monitored parameters is the number of reads per gene or transcript. This is set in the simulation phase by the expression profile. We have developed a list of metrics. C is the number of reads in the simulation stage, as C is the number of reads mapped by the tested alignment program. The proportional of lost reads and incorrectly mapped reads is reflected by the difference between these metrics, C and S. Correlation between them was also calculated. We estimate the proportional of genes or transcripts to which reads were mapped with minimal error. The normalized error range from -1 to 1 as calculated by the formula. The results of testing Illuminous mapping are shown on this slide. STAR took many times longer to run and used more memory than HiSight2 when mapping on genome, but the accuracy was similar. When mapping to transcriptome, Salmon came out on top. It had the best percentage of reads mapped with minimal error. When mapping the ONT platform reads, the Minmap2 alignment on the genome proved to the best. It shows fewer reads were lost and more reads were mapped with minimal error. Salmon was the worst performer in transcriptome mapping. This was due to a high percentage of lost read. When testing each program, the normalized error matrix was visualized as a histogram. A normalized error of 0 reflects the ideal work of the algorithm because the number of completed reads equal matched. When there are lost or incorrectly mapped reads, the normalized error shifts to negative and positive values. This is indicated by the left and right bars on the histogram. In terms of normalized alignment errors per genome, STAR and HiSight2 show excellent result. Salmon showed a lower percentage of lost read and more correctly alignment read when mapped on transcriptome. Salmon showed the lower percentage and Minmap2 more correctly aligned read when mapped on the transcriptome. Confirm the above, the mapping on ONT data to the transcriptome by Minmap2 and Salmon shows a high percentage of both lost and incorrectly mapped reads. In conclusion, we have shown that we can use simulation to assess the correctness of mapping algorithms. For Illumina genome mapping, HACCP2 should be preferred due to lower memory consumption. For quantification, should use Salmon. For mapping ONT reads per genome, Minimap2 should be used due to its good accuracy. Mapping reads to the transcriptome, as in the case of short reads, given unsatisfactory results. I would like to thank my colleagues from Institute of Biomedical Chemistry for taking part in this study. Thank you for your attention.
Thank you very much, Elizaveta, but I want to mention that you indicated that Illumina is not suitable for finding alternative splicing due to the short reads. In fact, because the reads are overlapping, alternative splicing can also be found in this data. However, it is believed that Oxford nanopore technology is more suitable for that task. That is just to comment. And Professor Firdaus, we move further.
Thank you. I invite Dr. Lau Tek Yew from University Kebangsaan Malaysia Medical Molecular Biology Institute. Dr. Lau, you have the...
Thank you very much, Prof. Firdaus. Okay, a very good afternoon to you all. I feel very honored to be invited to speak at this roundtable on bioinformatics between Russia and Malaysia. So today I'm going to present one of my recent projects, which is to use proteomics to search for microproteins in the colorectal cancer cell lines. And so before I start, let me explain about the background project, because I'm working in proteomics. And one of the questions that always bugged me is that why is it that data-dependent MS mass spec experiment usually only 20 to 50% of the MS2 spectra are matched to peptides in database searching, right? So if you look at the figure on the right, you have a series of B and Y ions on the MS2 spectra. This is an ideal case where most of the fragment ions, the daughter ions, are matched to the fragments of peptides. But in this topic has been discussed quite long ago by Mikalski and in a paper in Journal of Protein Research. And among the reasons that they give is that most of these can come from the poor quality of the MS2 spectra due to noise, contaminants, and some of these spectra even contain few peaks. And sometimes it can also contain mixed spectra, because sometimes when the isolation of the precursor happens, maybe 2 or 3 precursors can be co-isolated, and that result in the MS2 spectra that consists of daughter ions coming from 2 or 3 precursors. So the mixed spectra is hard to be disentangled until they develop an algorithm called a second peptide search. So sometimes the wrong precursor is selected for MS2. So then the mass, it doesn't match the mass of the supposedly the precursor. And sometimes also we have like experimental artifacts and sample contamination. For example, if you're growing as a human cell line, sometimes it's contaminated with mycoplasma. And when you search, you don't include mycoplasma protein. So some of these protein, the peptide actually match. Some of these MS2 spectra are actually coming from mycoplasma, but not from your desired sample. But some of the more sort of legitimate reasons that most of the sample that we run contains mutations, SNPs, and splice variant, and sometimes the spectra that actually we get, we are only matching to the reference genome or the reference proteome, but then sometimes it contains mutation SNPs, which doesn't match exactly to the sequence in the reference proteome, especially for SNPs. But of course, we seldom find SNPs because it's hard to detect the splice junction with mass spec, with the MS2 spectra. And sometimes also it could be due to the undetected post-translational modifications. So this problem has been solved. It's quite a big problem that has been solved by, is in the process of being solved by using the open search technology, that we set a very high tolerance for the MS1, and then we will let the algorithm choose maybe whether it's a phosphorylation, whether it's a ubiquitination, for example, by having a very broad MS1 mass tolerance. But today what I want to focus is about the unannotated proteins, which is microprotein is one of those unannotated proteins. So if you look at the UniProt database, actually only about 20,000 open reading frame has been curated by UniProt, and most of the time in a proteomic experiment, which is MS based, we only use this 20,422 protein for database searching, and we don't typically include all those unreviewed protein from Tremble, which is purely predicted by a computational algorithm. So the second question that forms background is how many unannotated proteins are hidden in the proteomic data sets, the experiment, that can we uncover them? So we know that the genome annotation is based on three main assumptions or three main criteria to distinguish true open reading frames and those noise. So first, open reading frame must have AUG start codon. Second is that a single open reading frame can only encode for one RNA transcript, which means that only one protein can be encoded. And most of the time, the open reading frame is defined as something that can encode the protein, but then it must be more than 100 codons or 300 bases. So these are the limitations set upon genome annotation, automatic annotation pipeline. But then recently, scientists start to question whether these assumptions are correct, because many of them actually found genes predicted to be non-coding, and they have demonstrated that these non-coding sequences actually they can encode protein or small peptide that are stable and active at the same time. So if you go read the latest paper about these dark proteomes, there are quite a lot of papers that describe this part of the dark proteome, which is which consists of open reading frames that are outside of this 20,400 for the human genome. Yeah. So this is a small review that I have co-written with my student. So this describes some of these sort of definition of these so-called unannotated protein or alternative open reading frames. One of those are those proteins that consist of less than 100 codons. And it could also be that the protein is encoded from the 5' untranslated region or the 3' untranslated region. Or it could be encoded by the typical ORF, but then by an alternative open reading frame, right? So this the second figure actually shows that there are different parts of the genome and also transcriptome that can actually encode for this so-called alternative open reading frame or alternative proteins. So yeah, this 5', 3', and also the so-called non-coding RNA, for example, circular RNA and the long non-coded RNA actually can also encode for open reading frame that actually in turn result in translation of stable and active proteins. So before we start the project, actually we go to the recent literature 2020, and because our institute is interested, we are very much focused in colorectal cancer actually. So then in that, we actually look for, you should read this paper, you find that some of these unannotated protein has been actually implicated in cancer development. So if you look at this, they are involved in different hallmark of cancer. Some of this protein, which is less than 100 amino acids, they activate the hallmark of cancer. Some of them actually inhibit the hallmark of cancer. But then for a number of hallmarks, actually, we do not find any unannotated proteins that play a role. So there's still blue seas that are out there that we can look for interesting proteins that might play a role in this cancer development. So in this project, actually, the project started in early 2022 by my master's student called Elisa. And in this project, actually, the main workflow we get our cell from within our institute, because we have the colorectal cancer cell line that come from different stages of cancer, the dukes A, B, C, and D. And also we have a normal cell line that we use as a control. And the experiment is very simple, where we grow the cell and we lyse the cell, and then we extract the protein. And we use acid precipitation and also molecular weight cutoff to only filter for protein that are less than 30 kD or 10 kD. And we proceed for reduction, alkylation, and trypsinization before we subject them to LC-MS analysis. And this part has been pretty much done by Elisa, but still analyzing the data. The second part involved building the database from these unannotated protein databases. There are currently quite a number of unannotated protein databases. for example, OpenProt, small protein databases, and SORF.org, and plus a few others. So these databases can be the data, for example, OpenProt, they are substantiated by data from mass spec, and SMProt and SRF, some of them are substantiated by data obtained from RiboSeq. So there are certain degree of evidence that is available for this, the assistance of this unannotated or this alternative open reading frame before we put into database. So I work with Elisa to combine this database using MATLAB, and at the same time also she also determined the mass distribution, frequency distribution of those alternative open reading frame, also the isoelectric point. And later on, since 2021, AlphaFold 2 has become popular, and we decided to work with Prof. Firdaus Raya from UKM as well. We plan to, upon identification of the small peptides, because it's unannotated, so it's very highly likely that the structure has not been solved as well. So we also try to look at what are the 3D structure of these unannotated proteins. So this figure shows the step that we use MATLAB to combine these different protein database like OpenProt, SRF.org, SMProt, and the recent data obtained by Sebastian van Tisch from the Netherlands, my collaborators, published in Nature Biotech. And also, of course, we include the conventional protein database from the UniProt that consists only of the SwissProt, which has these 20,000 fully annotated open reading frames from the traditional part of the human annotated genomes. And then these are different steps that we and Elisa perform to get a database, identify a sequence, peptide length, peptide mass, and isoelectric points. And also, from this, actually, we combine the different database. So we remove the redundant sequences, and we end up with like 633,000 of sequences, which a lot of them are predicted sequences, but without evidence from mass spec data or from the RiboSeq data as well. So so from these databases, we're interested to use this database to search for proteins that are present in the cancer cell lines, which we will do a bit later. Then, of course, this the mass distribution of the combined data set, of course. So we also perform a sort of theoretical calculation of the mass distribution of these unannotated proteins, and this just shows that the algorithm is actually used to match the known databases and see what is there any differences, and I think the mass differences is very small. So the algorithm actually works very well even for a very large protein like Thymine. So at the same time, about a month ago, undergraduate student actually joined my lab as an intern. So she will spend 3 months in my lab. So we are trying to see if we can identify this. The question is that in the first experiment, actually, we use this small molecular weight filter to enrich the small proteins. But in the second part, we'd like to see if without enrichment from the traditional data set, we can actually identify this protein or not. So we actually get this data set from the National Cancer Institute's CPTech project, or this Clinical Proteomics Translational Tumor Analysis Consortium. So this data are published in 2014 and 2019, and the 2014 data set consists of 60 60 proteomics experiment from stage 1 and to stage 4 of colorectal cancer tissue. And the second experiment actually consists of 99 proteomic experiment that is the proteogenomic analysis of stage 1 to stage 4 of female and male patients' cancer tissues. So during this search, we incorporate the combined database. And then what we found is that we are able to identify at a 0.1% false discovery rate. And we can identify about 9,000 proteins from this from the 99 sample data set, where we identified about 8,000 proteins as coming from the typical SwissProt databases, but there are also there are about 400 protein sequences that is coming from these unannotated or alternative open reading frames, and about 474 of them are purely predicted sequences, which based on ab initio prediction of genome annotation, and then above 75 and 257 has been previously validated by mass spec by 1 or 2 peptides, as well as the VHS are those sequences that are actually validated by ribo-seq experiment. So in this case, so we do. It means that from this large-scale proteomic data sets of cancer tissue that is deposited in the NCI, actually we can identify this so-called unannotated or alternative open reading frames from the patient's samples. So this slide actually. Okay, so this slide actually gives a little bit information about the localization of all these proteins. So some of these proteins actually coming from the coding sequences, but they are expressed from an alternative open reading frame. Some of them are expressed from the 3' UTR, 5' UTR, as well as some non-coding RNA as well. So we are still in the middle of trying to analyze further the data. And about these 647, about 300, they are actually smaller than 106. So yeah, and the next step, so actually we also perform, so these are the 5 different alternative proteins that we found. And if you see that actually some of them are encoded by the downstream open reading frame at the plus strand, and there are some that is encoded by the upstream open reading frame that's overlapping with the existing coding sequences, but is actually expressed, it was encoded by the negative strand of the DNA. So I think that's some of the preliminary results that we have at the moment. So actually, yes, we can actually detect these unannotated alternative proteins in existing MS data set. So there are more to go, and we are going to analyze more data sets. And these proteins are encoded by non-coding RNA, upstream, downstream open reading frame, and alternative ORF. And then actually, if you fractionate your sample enough, and there is no need for specific enrichment for these small open reading frames encoded proteins. And then we will proceed with more functional studies to study some of these so-called alternative protein in the future. Yeah, thank you.
Okay, thank you, Dr. Low, for that sharing on microproteins from CRC. Over to you, Professor Lisitsa.
Yes, okay. So I announce next speaker, Timur Shkrigunov. He is working as a junior researcher in the laboratory of protein pathophysiology, and he will report on combining proteomics and informatics approach for analyze keratinocytes. Timur, are you online?
Now I'm ready to start. Good afternoon, dear colleagues. It's a great pleasure for me to participate in such international rationalization seminar. And I'm from the Institute of Biomedical Chemistry from the Laboratory of Pathophysiology. And today I'm finishing the proteome block of today's seminar. And I will talk about our study on the proteomic characterization of cut keratinocytes exposed to surfactants. To start with, I'm just going to give a short introduction on surfactants. We had a hypothesis that surfactants cause cell damage to skin cells. We studied 2 surfactants, SDS, sodium didecyl sulfate, which is anionic surfactant, and trisone X100, which is non-ionic surfactant, but they're widely used in a liquid, paste, powder, and shaped cleaning agents such as shower gels, shampoos. They're widely used in the laboratories and so on. So the background was that there is no direct evidence supporting that such surfactants can be toxic to the skin, we use the subtoxic concentrations of SDS, and there is no direct evidence supporting that subtoxic concentrations of SDS and tritone can cause protein-protein interactions and biological processes perturbations in human epithelium. So it's known from the scientific evidence that keratinocytes have an essential and a big role in skin drug metabolism, and human Hakat keratinocytes are spontaneously immortalized in vitro keratinocytes from histologically normal human epithelium. So that was the object of our study. We used Hakat keratinocytes as a model of human epithelium. Regarding the surfactants concentrations, we used 25 microgram per milliliter of SDS and the same concentration for tritone. And in this slide, you can see the microphotographs of the pictures of Hakat cells exposed to such surfactant concentrations. A control, B SDS, and C is tritone. And we performed viability tests, and that showed that the cell viability doesn't decrease. Regarding the methods, we used our protocol for increasing the sensitivity of MS protein detection. It is the truncated SDS page protocol. You can see in this slide, we're loading samples in these holes. It is the apparatus used for SDS page. And as the proteins reach the border between stacking and running gel, this protein band was cut entirely and holistically for the subsequent liquid chromatography with tandem mass spectrometry. And in this picture, you may see our photos of gel. The red squares show the protein bands which we have cut for the mass spectrometry. And as you may see, the mass spectrometry data was processed using the Search GUI search engine with Xtandem and MSGF plus search algorithms. And we worked with initial data of protein reports of peptide shaker. And we speculate that for protein extraction, it's better to use detergents. And this protocol is very beneficial for detergent cleanup, the detergent elimination, because the detergent in the samples can interfere mass spectrometry analysis. So after this SDS removal, which the proteins will be gel bound, perfectly denaturated and ready for in-gel digestion, we developed this 1D gel concentration protocol. And the runtime in SDS page was only 15 minutes. So just to give an overall view of our results, we performed gene ontology enrichment analysis of proteomic data. And as you may see from this picture, in general view, even at subtoxic concentrations of SDS and tritone, xenobiotic detoxification pathways were significantly affected, were significantly upregulated, as you may see here, in Hakat keratinocytes. And it's just a general view, but we wanted to, as I call it, the eagle-eye viewpoint, to view more specifically to the interesting proteins, to the essential points of surfactants exposure. So that's why we developed comparative proteoinformatics pipeline, which permitted us to direct our attention to the very essential proteins. And as you may see here, in SDS exposed Hakat cells, we've seen the upregulation of MCM6 protein complex, which has a key role in the DNA replication because it's on both inverse DNA and progress the fork prolongation. And as you may see, 3 proteins from this family were overexpressed in SDS exposed Hakat cells. These proteins are associated with several diseases, including skin cancer. And as you may see here in the protein-protein interaction map, these MCM3 and MCM6 and MCM7 proteins were just joined in one protein-protein interaction map. And we performed the additional independent experiments in order to ensure the viability of 1D concentration. And here you may see the mRNA expression levels of MCM6 in SDS64 cells. The yellow bars show the mRNA levels, and these green red bars represent the mass spectrometry expression levels, expression assays of these MCM6 protein. And we speculate that MCM6 may be the checkpoint of the possible SDS-initiated proteomic landscape changes in human epithelium, and including these changes leading to the occurrence of even tumorigenesis, because according to the literature, this MCM6 protein was used as a biomarker of skin cancer. And regarding the return treatment, we indicated the identified proteins in this map. You may see the cytochrome 2C1, which the comparative protein informatics indicated these proteins as very important. And yes, we've detected it's an orphan cytochrome. Its content in skin is usually low or even undetectable in skin. And the 1D gel concentration protocol permitted to identify that. And that's why we made the conclusion that it's highly effective for the identification of low abundant and even missing proteins, because in our previous work, we were able to identify PSG7 protein, which has never identified before, but this 1D gel concentration protocol permitted to identify that. To conclude, just several remarks. According to the 1D gel concentration for the detergent elimination, it's very effective for the identification of proteins which are presented in very low levels in samples. You may see the link to the article here. And our main finding was that even at such non-toxic low concentrations of SDS and tritone, xenobiotic detoxification pathways in HCAT were significantly affected, significantly upregulated, and comparative protein informatics utilization permitted us to identify non-obvious features of surfactant exposure, as I call it, eagle eye viewpoint in our scientific reports article here. And as a general conclusion of our work, we conclude that HCAT offers a biologically relevant and biologically standardized because of the unlimited growth model that can be used to test drugs, to study disease mechanisms, and develop new treatments. So thank you very much for your attention. Thank you.
Thank you, Timur, for your presentation. And colleagues, once again, that we can use chat to ask the questions. And again, I would like to focus on the main aim of our roundtable. We somehow achieve it. We go forward it. We just have to understand that we are talking the same language, and we are exploiting the same problems, like, for example, 3D structure analysis, docking, prediction of medical plants, proteomics, MSMS analysis. So we just have to capture that we are on one page, and maybe we should not go deep into discussion of every paper, because it requires a specific conference format. Okay, just think about that, because we are ahead of schedule, but I think it's quite good because we have a very condensed agenda. Professor Firdaus, how do you think on that point?
Yeah, I agree, because we can use this platform as a more of an introduction as to what each other is doing.
Okay.
And we can find matches that are relevant to each other's interests. So I think
because if we go into details of each report, that will not allow to focus us on the whole agenda. So please present the next speaker, which is from Malaysia.
I invite Dr. Jenkit Tan from the Faculty of Medicine, University Kebangsaan Malaysia. Dr. Tan, you have the platform.
Right, thanks, Prof. Firdaus, for the introductions. So good afternoon, everyone from Kuala Lumpur. I'm Tan from the Faculty of Medicine, UKM. So I'm going to share some of my prelims findings from our laboratory on the fecal lipidomics and metabolomics profiling in aging rats. So aging is a progressive loss in physiological functions, and the hallmarks are including like genomic instability, mitochondrial dysfunction, to name a few. Metabolic alteration is often associated with aging, but to be more complicated, our metabolism is affected by, besides endogenous like genomics factors, also affected by environmental factors, for example, diet, physical exercises, physical activities, and also lifestyle. So in this case, we are using a lab model, the males bred dolly in a controlled environment, which is a lab, to find for the age-associated lipids and metabolites that are associated with aging. So our methodology is to collect the fecal samples from, this is a longitudinal study for over 2 years. So we are collecting the fecal samples every 6 months and isolating the metabolites and lipids, and acquire the data using the LCMS and doing the typical processing steps, data pre-processing, also the statistical pathway analysis, and as well as the enrichment analysis. This is our prelim pre-processing data. So we detect more lipids features than the metabolite features. More than tens of thousands of lipid features have been detected, and around 800 significant lipids have been detected. For the polar metabolite, more than 2,000 total metabolite from both positive and negative ion modes has been detected, and this finally correspond to over 100 significant metabolite associated with aging. So by using unsupervised multivariate analysis, partially squared discriminant analysis, PISDA, we can see both in the positive and negative modes. In the positive mode, the lipids separation, there's a trend from 6 months to 24 months. And while the separation for the negative mode, it is more separate in two groups.
Excuse me, Professor Tanjan Keat, sorry for interrupting you. I have an urgent question. Which type of mass spectrometer have you used? Because I have skipped it maybe. Which type of mass spectrometer?
We are using the Orbitrap mass spec.
Orbitrap of which model?
Q Executive High HF.
HF. Thank you, because for me to understand the topic is important to keep in mind the type of equipment. Sorry for interrupting.
Yeah, no problems. Okay, so you can see for negative mode there is like a clear separations before the 12th month and after the 12th month. So based on the trend, we can see these significant lipids are related to phosphoglycerol lipids, but we also surprisingly detected some of the fatty acids, and these are correspond to unsaturated and the branched-chain fatty acids. For the metabolic data, it is more simplified. The trend is still similar. You can see the separations from 6 months to 24 months. There's a trend, okay, every 6 months, and the separations for the negative modes still before and after 12 months. And these significant metabolites correspond to various metabolic pathway. But to summarize, they are related mainly to amino acid metabolisms, and they are decreased with age. So this is a prelim study. We have also collected the cognitive functions, the muscular functions of the animals, and we would like to correlate these significant lipids and metabolites with all these physiological functions, and finally to come out with how they are actually associated with aging and the hallmarks of the aging. So this is a short presentation. So I would like to thank my staff, Halida and Nazira, for collecting and maintaining the animals and fecal samples for 2 years, and also my funding agency, Ministry of Education, Malaysia, and we are the LCMS laboratory in the Faculty of Medicine, and we receive samples from Malaysia across, various institutions across the Malaysia. So yeah, thank you so much for your attention.
Thank you for your report. We just have mentioned that we don't ask questions, but I will take my role of co-chair and ask you, what was the time of the HPLC gradient?
We use 22 minutes gradient.
22 minutes, both for negative and for positive was the same?
Yeah, we use the same gradients, yeah.
And do you use the standards?
Yeah, it's a good question. Yeah, for lipids, we do use the standards. We use the commonly the Avanti standard. It's a mix of standard of 14 heavy isotope label. But for metabolic study, we didn't use it because we couldn't find a community, a standardized, difference. We still don't have a standardized internal standard for metabolic studies. Some study, they might use different sets of standards, and some labs, they use different sets. So we still do not have a consensus on the metabolic internal standards. So I think this one, we can look into it and develop a community agree on what kind of standard we should use for a metabolic study.
Okay, thank you very much. Sorry for asking question, Professor Firdaus. I have seen that you were not online, and therefore, okay, we continue the topic of metabolomics, and Anna Kozlova, senior instructor from Center of Practical Education, which is in Moscow, would report on some programmatic tricks that she has done, but by using direct MS. Anna?
Thank you for introducing me. Good afternoon, dear colleagues. Thank you very much for inviting the table on bioinformatics. I'll start my presentation. Well, I work with different types of biological data information, and I work with genomic data and transcriptomic, proteomic, but my favorite scientific field is analysis of metabolomes and metabolomics in general. So my report focuses on the method for reanalyzing direct injection mass spectrometry data and answered the question, what open source programs can be used for this purpose? There are several analytical approaches in non-targeted metabolomics, such as liquid chromatography coupled with mass spectrometry, gas chromatography coupled with mass spectrometry, flow inlet analysis, and others. The usual method is direct injection mass spectrometry, also called DIMS. This method does not require any chromatographic separation of metabolites in the sample prior to mass spectrometric identification. The samples free of proteins, so in this Eppendorf contain only metabolites, using glass syringe, fed direct into the ion source of the mass spectrometer. Since DIMS is used for screening purposes of the large number of blood plasma samples, there has been a need to automate the processing of mass spectra of these samples. All existing approaches for processing direct injection mass spectra based on the commercial programs such as MATLAB and Compass data analysis, and common user who is not the operator of mass spectrometer just haven't got these programs on his personal PC. So there is need to find the open source packages. And there is the problem. We suggest the pipeline based on open source packages for our thoughts. And to confirm the possibility of using selected programs, we compared our results with previous results of analysis of the data which was obtained in the Laboratory of Mass Spectrometric Metabolomic Diagnostics, part of Institute of Biomedical Chemistry. I'll start with initial data. Previously, mass spectra of blood plasma samples were obtained in IBMC laboratory from 100 volunteers with different health conditions. The study included 20 healthy volunteers, 20 overweight volunteers, and 20 volunteers for each stage of obesity. Blood plasma samples free of proteins as I mentioned above, they contains only metabolites, were fed into the ion source. In this study was used Bruker-Max's impact mass spectrometer. For each sample were obtained mass spectra. So one sample was scanned with 60 mass spectra. So one sample was characterized by a series of 60 mass spectra. The mass spectra obtained on the instrument were automatically recorded in the D directories. The D directories contain binary files with information about MZ values and intensities. So there are 60 mass spectra. Using MS Convert as a part of ProteoWizard platform, we convert D directories into MZML files. MZML is usual XML files with information about mass spectra. To process mass spectra with an open source package MultiQuant, we need to have one spectrum for one sample. So using MSN-base package, we obtain the consensus mass spectra. It means that all 60 mass spectra are combined with some rule. Only those peaks were left, the MZ values of which were within the specified mass range and had the maximum intensity. As a result, we get one mass spectrum for one sample. Next, we use standard MultiQuant pipeline to obtain mass lists for each sample. This standard pipeline contains several steps as a baseline estimation, baseline correction. That means that all intensities in this mass spectrum are on the same base level. After baseline correction, we prepare peak detection. And the important step is the peak alignment across all mass spectra or from all samples. And after peak alignment from each mass spectrums, we obtained the mass lists. Finally, using Mummy-Choc algorithm, we annotate MZ values from mass lists. Compared to the other algorithms, Mummy-Choc is not based on metabolic pathways, but on metabolic modules. Metabolic modules are larger networks of interactions than metabolic pathways. Mummy-Choc doesn't routinely identify metabolites. Based on unbiased statistical tests, Mummy-Choc allows you to determine the most likely profiles of metabolites that make up the metabolic pathways through using your feature table obtained on the previous step. Well, comparing our results with the previous results, we found that the MultiQuant and Mummy-Choc approach made it possible to annotate a larger number of metabolites compared to the proprietary one. In addition, our approach identified all nodal metabolites in the metabolic pathway of steroid hormone biosynthesis according to KEGG knowledge base. Also, pathway analysis revealed all the same metabolic pathways that were previously presented in previously analysis. So we can conclude that our approach are reproducible and can be used for other users. Thanks for the IBMC teams, administration, the Laboratory of Mass Spectrometric Metabolomic Diagnostics for share the data and the results. And thank you for the team of Post-Genomic Data Analysis Laboratory. I appreciate your attention and your time. Thank you for your attention.
Thank you very much, Anne, for your report and for your work. I just want to comment that the main idea, which was not very transparent, but afterwards it's on the surface, that direct infusion mass spectrometry data appear to be quite equivalent to multi-TOF data, therefore means the direct infusion in electrospray mode can be processed with the same programming arsenal which are available for our language, which previously has been developed for analyzing MALDI-TOF, and the program MALDI-Quant is that exists maybe already for 10 years. And has shown that this program can be applied not only for MALDI data, but also it can be used to process the direct infusion MS data, and that was quite amazing and not expected because normally people are doing it in another way, but specific software for DIMS data processing does not exist to my knowledge. Okay, Professor Firdaus, I give the floor to you for announcement of the next talk.
Thank you. For the next speaker, we have Dr. Ahmad Madiani from the Institute of Systems Biology, University of Malaysia. Dr. Madiani.
Thank you very much, Prof. Firdaus, and thank you for inviting me to this interesting roundtable conference. Please, can you let me share my screen? I want to share with you my research about metabolomics in natural product research. My name is Ahmed Madiani, as Prof. Firdaus mentioned. I am from Symbiosis UKM. Okay, as all you know, that most of the drugs that they are based from natural products, and we have many drug leads from these natural products, as from plant, as from marine drugs and other resources. And Malaysia, it has biodiversity resources, which is estimated 15,000 plant species and other resources. As from that, I was interested and I selected few plants for my study, as I am focusing on anti-diabetic from medicinal plant, as I have few example of plants, Phyllanthus nerori, Phyllanthus acidus, Neptunia, as my current project on Neptunia oleracea and Phyllanthus acidus. Today, I will focus on Phyllanthus nerori for anti-diabetic on in vivo model. And then before we proceed with my study, I just want to share with you a few slides about when you want to develop a drug, we need to focus on the safety, the efficacy, the quality, and the consistency. All these parameters or characteristics can be studied using the metabolomics. And this method is very advanced, and it has potential in natural product research nowadays. And it is accurate identification and quantification of metabolite in the biological system, which is the plant or the cell or animal cell, organ, anything. As compared with other omics, metabolomics is, we cannot say it is the best among them, but it is, we can use it as complementary with each other. Then before we proceed, we need to standardize the plant extract, as we need to control few factors, which is the growth condition. We cannot use the pesticide or insecticide contaminant. And also we need to choose the geographical location, harvesting time, the plant part, and the drying and extraction method. We need to optimize all these parameters because at the end it will affect the final product before we can proceed with the animal study. As I did, I plant my own sample to minimize the effect of these factors. And I have studied the growth stages for the Phyllanthus nerori, and then the drying methods, the solvent, and also the extraction methods. I optimize those factors, and as I published before in this research. In here I have 3 growth stages, which is 8, 10, and 12 weeks, which is the flowering stage, and 2 weeks before the flowering stage, and 2 weeks after the flowering stage. And I found that the flowering stage, it is the best stage which can produce most of the metabolite, which is contributing to the bioactivity, DPPH, alpha-glucosidase, and other biological activities. We choose the 10 weeks. For the drying methods, we compare between the air drying and the freeze drying and oven drying. We find that the freeze drying has the conserve most of the metabolite of the plant and can show those metabolite has the contribution to the bioactivity. After we choose the growth stage, the drying method, and the extraction, we proceed to do the standardization of the plant extract. We do the HPLC for few metabolite, the one we identify in the plant, and we quantify them. And before we proceed with the animal study, because we choose 2 different doses, 250 and 500, I quantify those metabolites in these 2 doses. And then I proceed with the detection of biomarkers in the diabetic-induced rats. This is my design. I have 2 different groups in the beginning. I have normal diet and the high-fat diet. I induce the obesity for 3 months, for 12 weeks. Then I proceed with the induction of diabetic with the STZ after they are significantly obese. Then we proceed with the treatment with, I have high dose and low dose. Low dose is 250 and the high dose is 500. And I have the positive controls as the metformin. This is when we check the obesity. After 12 weeks, we can see the significant difference before we proceed with the diabetic induction. After the treatment, we can see that we check few biochemical parameters, and we find that the high dose and the low dose of the phyllantha extract reduce the LDL and also the cholesterol, triglyceride, and also the glucose and the creatinine also, as compared to the metformin. For the metabolomic study, we see that based on the trajectory, we can see that the treatment with the both dose, low dose and high dose, can shift the rest to become to the normal, but they are still not normal because they have a few changes in the metabolite. And here we can see that here the high dose has the best effect and the highest effect as compared to the low dose. Here is the diabetic rats, and this is the normal. We can see that the high dose is shifting towards the normal rats. We can say that there is effects of this extract. And we show what are the metabolites, they are significantly affected with the treatment. Among them, we can see the glucose, it is lower after we give the treatment. Because here it is high in the obese diabetic. After the treatment become decreased, which is down-regulated. And few taurine, choline, glucose, and creatinine, they are down-regulated with the treatment. And other metabolites, they are up-regulated with the treatment. From that, when we see the metabolites, the one they are significantly different from the groups, we come out with the pathway analysis, which is involving all the metabolites, the one they are significantly affected. And we see the pathway, the one they are affected. And we discuss those pathways, and we see that the synthesis and degradation of ketone bodies, it is the main affected pathway. Then after that, we see all the one affected here, all the pathway which is involved, the one they are significantly and not significant. And to summarize, we need to come out with our own design for the pathway, and which one is the one affected and the most significantly affected. At the end, I want just to acknowledge the Symbiosis and UKM and my previous institute in UP and my supervisor also. And in Symbiosis, we have a group of experts, which is proteomics, metabolomics, bioinformatics, and transcriptomics. If you want to collaborate with us, we have all these experts in one place. You are welcome to collaborate with us. Please feel free to contact any of us. And I can share with you this. We have a upcoming conference on December in Malacca. You can check our website and please join us. You are welcome to come to Malaysia. Thank you very much.
Right. Thank you, Dr. Ahmad. We've basically reached the last of our speakers scheduled for the day. So I think, shall we open for questions if there are any, to go back all the way through, Andre?
Yes, formally, but I think that it's more important, colleagues. If someone do have questions, it is right time to ask them. But as I understand, some of the questions were already in the chat, and it is quite important to digest all the information and after that make targeted, I would say, contacts. And what is more important, I would like to ask you and Professor Paroykov to talk kind of summarizing what are the summarizing in a very general way what could be the contact points, what directions could be interesting for at least exploring an opportunity of collaboration. I give the floor to Professor Paroykov to summarize, and then I will ask you to do the same and to make an amendments or additions. Professor Vladimir?
Okay, thank you very much, Andrei and Professor Firdaus. Yeah, actually, we listened to 15 talks, and they actually represented wide areas where I believe people could match their research one to another. Yeah, for us, for Nikita and me, of course, a talk of Dr. Bazar and Dr. Medliani were most interesting, yeah, because they also deal with medicinal plants, yeah, and phytocomponents. I think that we have some possibilities for future collaborations. I do believe that between some other participants of our roundtable, also maybe such pairing, and to make it more efficient, it seems that the organizers should distribute the abstracts and maybe somehow the presentations between the participants, yeah, to think a little bit more, and of course to have the email addresses to contact directly the persons with whom we could collaborate. I should also say that, of course, it seems that both Malaysian and Russian scientists have some deficit of interactions, because we probably know good people or researchers from United States, from England, from some other big countries, but not between Malaysia and Russia. So it was very, very useful to get acquainted to some research which are done in those countries. I think it's not just my opinion, but also general opinion. So thank you once again, Andrei and Professor Firdaus, for organizing this very interesting roundtable. And let's hope that we will collaborate in the future. Thank you.
Okay, thank you very much, Professor Firdaus. Could you conclude our meeting?
Thank you, Andrei and Professor Paroykov. I do agree in the sense that I see some complementary aspects of what has been presented. It's clear just now, I think Nikita and Dr. Noraza, there are some who are doing more of the experimental discovery of the compounds, and then you're also doing the databasing and compiling of that. So that's very complementary in a sense, and that's a clear way of how they can work together. Myself, I am quite interested with what Dr. Oksana was presenting just now, and it's got some aspects that are relevant to my work as well. And I think it's clear that the metabolomics also we have clear avenues of complementing the groups from Malaysia and Russia. So I also echo what Professor Paroykov mentioned just now. We have been collaborating with a lot of other groups from the West, Japan, Australia, but I don't think we have had many or any collaborations with any Russian groups to my knowledge. Despite the fact that we actually have I think students, medical students, and who actually go to study medicine in Russia. So I think this is an opportunity for us to take this forward. And I also agree with what Professor Paroykov was saying, that perhaps we can compile the abstracts and the context for the person so that the individuals can follow up. And perhaps we at the secretariat or organization level, along with Neelam, can also do some follow-ups as to what's going on and how they are interacting, just to keep track of whether things are actually happening, or people have just sort of died down after this meeting. Okay, so I think that's more or less what I can summarize, because it's a lot of diverse presentations just now. So to summarize every single one would be, I think, a bit difficult. But I think I see clear directions as to how some groups can interact and can continue, even perhaps with just starting with the workshop Dr. Firdaus was mentioning just now. So that would be great as well. So I think that's it from me, Andre. Over back to you.
Oh, okay. Thank you very much. And in conclusion, I thank all the speakers for preparing and presenting their materials. It was very, very interesting and important for all of us. And certainly I thank to who were organizing the event from Malaysia side. Neelam, Rani invested very much efforts, and Maya from Russian side, and also I would like to mention that some of the speakers, young speakers from today's seminar, they will visit UTM in May, and they will be organizing some event where we can return back to the topics and in more details to discuss the prospects for collaboration. Thanks to everyone. And I would like to ask Ahmed Mediani to send the link to the event that he has announced. And I ask organizers to distribute all the materials and to save our chat and the questions. And so have a good day. And there is a rare experience when we are not out of the schedule.
May I say something, Dr. Lisitsa?
Certainly. Go ahead.
Okay. Thank you very much for participating in today's roundtable. As what Professor Oroykov and Professor Lisitsa has mentioned, we are already collaborating with UTM, and we look forward to collaborating with UKM as well. And moving forward, I think we should follow up from here to the relevant people that can exchange ideas in the future. And moving forward, like what Professor Lisitsa has mentioned, his young researchers are basically coming to Singapore to attend a seminar which was actually recommended by Dr. Lau when the Russian delegation was in UKM. And on the 11th of May, we are also going to UTM to present a one-day presentation by them. And we hope that this is just the beginning of both sides collaborating and doing research, because I think there's a lot of things that we can learn from each other.
I see Professor Zaitun there. Do you want to say anything, Professor Zaitun?
Yeah. Very good afternoon. Yeah, so I just rejoined because I have another engagement, but I think Firdaus has said have said on behalf of UTM just now. So I'm looking forward to the visit by the Russian students in May. So we look for more further collaborations between the Russians and us, inshallah.
Good, thank you very much.
Most welcome.
And before we leave, is it possible for everyone to on their cameras and we do a group photo as usual so that we can have some records of this roundtable meeting, please?
Okay. Thank you so much.
Did you manage to take all our photographs?
Yes. Thank you.
Okay, thank you so much for participating, and selamat berbuka puasa for all the Muslims who participated. I know it was a long day for all of you, and have a good Easter tomorrow for all the Russians.
Thank you to everyone.